something extraordinary begins to happen. Something that will take generations, thousands of generations, hundreds of thousands of years of quiet incremental change, but something that will ultimately produce creatures so strange, so wonderfully bizarre, so perfectly and exclusively suited to their isolated worlds that they seem to belong to a different planet entirely. This is the story of the island rule. This is the story of what happens when life is cut off, cornered, compressed, and forced to reinvent itself from the inside out. This is the story of giants that shrank, dwarfs that grew enormous, fearless birds, miniature humans, and mice that learned to hunt creatures 1,000 times their ancestral ambition. This is the story of evolution working not across vast open continents where millions of individuals compete across sweeping landscapes, but in tight, pressurized, resource-limited pockets of land surrounded by water, where the rules of natural selection get rewritten, sometimes dramatically, sometimes with breathtaking speed. And perhaps most importantly, this is a story that is not finished. The island rule is not ancient history tucked away in fossil records and museum display cases. It is alive right now on islands scattered across every ocean on this planet. It is happening in real time, and understanding it has become one of the most urgent priorities in modern biology, ecology, and conservation science. So let us begin. Let us travel to these isolated worlds. Let us meet their extraordinary inhabitants. Let us understand the invisible forces that shape them. And let us ask the question that sits at the very heart of this documentary. What does it truly mean to evolve in isolation? To understand the island rule, we first need to understand what makes an island different from any other environment on Earth. At its most basic level, an island is simply a piece of land surrounded by water. But that simple definition conceals an almost incomprehensible ecological complexity. Islands come in every conceivable size, from tiny coral outcroppings barely large enough to support a single palm tree, to massive landmasses like Greenland or New Guinea that dwarf many countries. They exist in every climate zone, from the frozen subantarctic to the scorching tropics. They are separated from mainland environments by distances ranging from a few kilometers to thousands of miles of open ocean. But what all islands share, regardless of their size or location or climate, is a fundamental condition of isolation. And isolation, in evolutionary terms, is the most powerful force imaginable. It is the knife that cuts a population off from the gene pool of its mainland relatives. It is the closed door that keeps new competitors from arriving and old predators from following. It is the pressure cooker that concentrates evolutionary forces until they become impossible to resist, until they bend and shape and ultimately transform even the most deeply ingrained biological traits. Think about what happens the moment a small group of animals becomes isolated on an island. It might happen dramatically. A volcanic eruption creates a new landmass, and a handful of birds or insects or reptiles colonize it. It might happen gradually. Sea levels rise over millennia, separating a formerly connected peninsula from the mainland. It might happen by accident. A pregnant lizard drifts on a mat of vegetation across open ocean and washes up on a shore where no lizard has ever set foot before. However it happens, the result is the same. A small founding population, carrying only a fraction of the genetic diversity of the mainland species, finds itself in a completely new environment with a completely new set of challenges and opportunities. Now, evolution does not think ahead, it does not plan, it does not make decisions. It is a blind process driven by a single simple mechanism. Individuals that are better suited to their environment survive and reproduce more successfully than those that are less suited, and over time, the traits that make them more successful become more common in the population. That is it. That is the entire engine. But when you combine that simple engine with the extraordinary and specific pressures of island life, limited resources, no large predators, new food sources, different competitors, the results can be genuinely astonishing. The island rule, as scientists have come to understand it, describes a predictable pattern that emerges from these pressures. Large animals that arrive on islands tend, over evolutionary time, to become smaller. Small animals that arrive on islands tend to become larger. It is not a universal law with zero exceptions. Biology is rarely that clean, but it is a pattern robust enough to have been observed on islands all over the world, across dozens of taxonomic groups, over millions of years of fossil evidence and thousands of years of observed ecological change. It is one of the most consistent and fascinating patterns in the entire history of life on Earth. The principle was first formally articulated in the 1960s by a biologist named J. Bristol Foster, who noticed while surveying island mammal populations that this size-shifting pattern kept appearing. He called it Foster's rule at the time, though the broader scientific community eventually settled on the term island rule. Foster had observed that island populations of large mammals, deer, elephants, hippos, certain primates were consistently smaller than their mainland counterparts, sometimes dramatically so. Meanwhile, small mammals, rodents particularly, tended to be larger on islands than on the mainland. He suggested that these changes were driven by ecological pressures specific to island environments, and subsequent decades of research have largely confirmed and refined his original insight. But why? What is actually driving these changes at the level of biology, physiology, and natural selection? To understand that, we need to think carefully about what life on an island actually demands of the creatures that live there. For large animals, the primary challenge on an island is almost always the same: food. A large body requires a large amount of energy to maintain. On the mainland, large herbivores like elephants or deer can range across vast territories, following seasonal food sources, moving through diverse habitats to meet their nutritional needs. On an island, that option does not exist. The land is finite, the resources are finite. An elephant that needs to consume hundreds of kilograms of vegetation every day cannot simply walk in a different direction when the local food supply runs low. It is stuck. And so the pressure on large-bodied island populations is relentless and consistent. Smaller individuals, who require less food to survive, do better. They produce more offspring because they are not perpetually stressed by nutritional deficiency. Their genes, including whatever genetic variants contribute to smaller body size, become more common with each generation. Over thousands of years, the average body size of the population decreases, sometimes by a startling amount. For small animals, the logic runs in the opposite direction, though it is equally compelling. On the mainland, small creatures like mice or rabbits or small lizards live in a world saturated with predators. Hawks circle overhead, foxes nose through the underbrush, weasels thread through every crevice. The pressure of predation keeps small prey animals at a disadvantage when they grow too large. A larger mouse is more visible, slower to escape into a burrow, and more energetically costly to maintain. Mainland mouse populations, shaped by millions of years of predator pressure, are optimized for smallness and speed. On an island without those predators, everything changes. The mouse that happens to be slightly larger than average does not pay the price for that size. It is not picked off by a hawk before it can reproduce. Instead, it may actually benefit from its size. A larger mouse might be able to defend better territories, access more food sources, or simply have more energy reserves to survive periods of scarcity. Larger body size, once a liability under predator pressure, becomes a neutral or even advantageous trait. And so, generation by generation, the population drifts upward in average body size, unconstrained by the predatory forces that kept it small on the mainland. This is the basic architecture of the island rule. It is elegant. It is logical, and it is supported by some of the most spectacular examples in the entire fossil record. Let us start with what is perhaps the most iconic example of island dwarfism: the dwarf elephants that once lived on islands scattered across the Mediterranean Sea and the Southeast Asian archipelago. The story of these miniature giants is so extraordinary that when paleontologists first began finding their fossils in the 19th century, they assumed they had discovered something entirely new, some unknown, unrelated creature. It took careful anatomical analysis to reveal the truth. These were elephants, real, genuine elephants. Just elephants that had been squeezed, generation by generation, by the relentless logic of island life into something that in some cases stood barely a meter tall at the shoulder. The Mediterranean dwarf elephants are particularly well documented. During the Pleistocene epoch, the period stretching roughly from 2.5 million years ago to about 12,000 years ago, sea levels fluctuated dramatically as glaciers advanced and retreated across the northern hemisphere. During glacial maxima, when enormous amounts of water were locked up in ice sheets, sea levels dropped significantly, sometimes by more than 100 meters. Islands that today sit surrounded by deep water were during those periods accessible from the mainland. Animals could walk to them, and many did, including ancestral elephants, the relatives of the modern African and Asian species that were themselves considerably larger than their living descendants. When sea levels rose again at the end of glacial periods, the elephants that had walked to these Mediterranean islands found themselves stranded, cut off, isolated, and the island rule began its patient work. On Cyprus, paleontologists have found fossils of Elephas cypriots, a dwarf elephant estimated to have stood about 1.5 meters at the shoulder and weighed perhaps 200 kilograms, roughly 4% of the body weight of the mainland elephants from which it descended. 4%. In evolutionary terms, the Cyprus dwarf elephant had shrunk to something approaching the size of a large pig. On the island of Tylos, near Rhodes, fossils of another dwarf elephant species have been found that survived until as recently as 4,000 years ago, making it one of the most recent survivors of Mediterranean island megafauna. On Malta and Sicily, yet another dwarf elephant species, Elephas falconeri, is known from extensive fossil deposits. Estimates suggest it stood only about 90 centimeters at the shoulder and weighed perhaps 250 kilograms. These were animals that a modern human adult could have looked down on. The rate at which this dwarfing occurred, as best as paleontologists can reconstruct it from the fossil record, was actually quite rapid in geological terms. Modeling studies based on the known ages of the island populations and the sizes of the founder animals suggest that most of the body size reduction could have occurred within a few thousand years, which, when you consider that we are talking about generation times of 10 to 20 years per elephant, represents only a few hundred generations. Evolution, when the pressure is sufficient and consistent, can work with remarkable speed. But the Mediterranean is only one chapter in the story of dwarf elephants. In Southeast Asia, a related but distinct evolutionary experiment was playing out across the islands of the Indonesian archipelago. Here on the island of Flores, the same island that will appear later in this documentary in a different, even more astonishing context, paleontologists have found evidence of a dwarf stegodon, Stegodon florensis, a relative of the elephants that was itself subject to island dwarfing. Fossils suggest it stood perhaps only 1.5 meters at the shoulder, reduced from mainland ancestors that were substantially larger. Flores, as we shall see, was an island with extraordinary evolutionary ambitions. It was not content to dwarf just one kind of megafauna. What these dwarf elephants tell us, beyond the obvious narrative of shrinkage, is something profound about the mechanics of island evolution. They tell us that even the most fundamental biological characteristics, the body plan that has been honed by millions of years of mainland evolution, are not fixed, they are flexible. Given the right pressures, given isolation and limited resources and enough time, evolution will rebuild almost anything. It will take a creature that has been one way of living and patiently, incrementally, transform it into something almost unrecognizable. This is one of the deepest truths that islands have to teach us. Now let us turn to the other side of the island rule, the gigantism side, because the story of island giants is every bit as fascinating as the story of island dwarfs, and in some ways even more dramatic. The principle, as we established, is that small animals on islands, freed from the predation pressure that kept them small on the mainland, have the evolutionary freedom to grow. And grow they have, in example after spectacular example across the world's islands. Rodents are perhaps the most consistent practitioners of island gigantism. The Coryphames buhleri of Timor, known only from fossils, was a giant rat estimated to weigh perhaps 6 kilograms, orders of magnitude larger than the house mice and small rats that typically colonize islands. On the Canary Islands, the prehistoric giant rat Canariomys brevoi reached similarly impressive proportions. In the Caribbean, several species of hutia, large rodents related to guinea pigs, evolved to enormous sizes on islands where predator pressure was relatively low. The largest known prehistoric Caribbean rodent, Amblyriza inundata from the island of Anguilla, may have weighed as much as 200 kilograms, making it roughly the size of a modern black bear. But rodents are not the only small creatures to exploit island freedom for gigantism. Reptiles are extraordinarily consistent practitioners of this rule. The isolation of islands has produced giant tortoises on the Galapagos, on Aldabra, on islands across the Indian Ocean and Atlantic, giant iguanas in the Caribbean, giant skinks that are now mostly extinct but whose fossils haunt the archaeological record of Pacific islands. And of course, most famously and most dramatically, a certain enormous lizard on a certain Indonesian island that we will examine in considerable detail very shortly. But before we get to the dragons, let us spend a moment thinking about why reptiles are so prone to island gigantism. Part of the answer is physiological. Reptiles are ectotherms. They regulate their body temperature through environmental heat rather than generating it internally as mammals do. This means that a large reptile does not face the same exponential increase in metabolic cost that a large mammal faces when scaling up its body size. A giant tortoise can, in lean times, slow its metabolism dramatically and survive on almost nothing. A giant lizard can bask in the sun for warmth rather than burning through calories to maintain a constant body temperature. This metabolic flexibility makes reptiles especially well suited to the boom and bust resource cycles typical of island environments, where food can be abundant in some seasons and desperately scarce in others. There's also the predator release factor, which hits reptiles particularly hard in island settings. On most mainland environments, reptiles, particularly lizards and tortoises, are constrained in body size by the presence of large predators that hunt them. Crocodilians, large cats, raptors, and other carnivores put a ceiling on how large a turtle or a lizard can realistically grow before its size makes it more visible, more energetically costly to escape. On islands without those predators or with significantly reduced predator pressure, that ceiling is lifted. Reptiles can grow as large as the food supply and their own biology will allow. And given reptilian metabolic efficiency, that can be very large indeed. The Komodo dragon, Varanus komodoensis, is the living culmination of this evolutionary logic, and it deserves the extended attention we are about to give it. The Komodo dragon is the largest living lizard on Earth. A large adult male can exceed 3 meters in length and weigh more than 80 kilograms. It has a thick, muscular body covered in armored osteodermal scales. It has a long, forked tongue that functions as an extraordinarily sensitive chemosensory organ, capable of detecting the scent of blood from several kilometers away. It has serrated, laterally compressed teeth designed for tearing flesh, and it has in recent years been revealed to possess a form of venom delivered through glands in its lower jaw, a discovery that overturned decades of scientific assumption that the dragon killed through bacterial infection alone. The Komodo dragon lives on 5 islands in the Lesser Sunda chain of Indonesia: Komodo, Rinca, Flores, Gilimatang, and Padar, though the population on Padar is thought to have gone extinct. Its range is tiny by the standards of any apex predator. The total area occupied by wild Komodo dragons is roughly 1,000 square kilometers, less than the area of many major cities, and yet within that tiny range, it is utterly dominant. It is the apex predator, the top carnivore, the creature around which the entire ecological web of these islands is organized. How did such a creature come to exist? The evolutionary history of the Komodo dragon has been the subject of considerable scientific debate and investigation. For a long time, the conventional wisdom was that the Komodo dragon evolved in isolation on its Indonesian islands, that it was a product of island gigantism, a small ancestral varanid lizard that grew to enormous size precisely because of the island conditions we have been discussing. This was a compelling narrative, and it fit the island rule neatly. But recent fossil evidence has complicated and enriched the story considerably. Fossil varanids, monitor lizards, the family to which the Komodo dragon belongs, have been found in Australia dating back millions of years, and some of these fossils are of impressive size. The Megalania, or Varanus priscus, an extinct giant varanid that roamed Australia until perhaps 40,000 years ago, may have reached lengths of 5 to 7 meters, making it the largest lizard known to have lived. The discovery of fossils closely related to the Komodo dragon in Australia, from deposits much older than any Komodo fossils from Indonesia, suggests that the dragon's lineage may actually have originated in Australia and later dispersed northward into the Indonesian islands, not the other direction. What this means for our understanding of island evolution is nuanced. The Komodo dragon may not be a case of a small lizard growing large in island isolation. It may instead be a case of a large lizard lineage that colonized islands and subsequently maintained, and perhaps refined, its large body size within the specific ecological niche of those Indonesian islands. Or the truth may be more complex still: multiple colonization events, populations that shrank and then regrew, extinctions of larger relatives that left the Komodo as the sole survivor of what was once a more diverse guild of giant varanids. What is not in doubt is what the Komodo dragon has become within its island environment. It has become the perfect island apex predator, large enough to take down prey as big as water buffalo, yet metabolically efficient enough to survive long periods of food scarcity, reproductively flexible enough to reproduce parthenogenetically without males in extreme conditions, and physiologically adapted to a specific suite of island prey animals in ways that no mainland predator has been forced to match. The Komodo dragon hunts using a combination of patient ambush and active pursuit. It will wait beside a game trail for hours or even days, completely motionless, until a deer or a goat or a pig passes within striking range. Then it explodes into action with surprising speed. A large Komodo can reach speeds of 20 kilometers per hour over short distances and delivers a devastating bite to the prey's throat, legs, or underbelly. The combination of anticoagulant compounds in its venom, its serrated teeth, and its sheer physical power often results in serious injury to prey even if the initial attack does not produce an immediate kill. The dragon's extraordinary chemosensory system allows it to track wounded prey animals for kilometers, following the scent of blood until the animal eventually collapses from blood loss or shock. Within the ecological context of Komodo Island and Rinca, this hunting strategy has been refined over millions of years against a specific suite of prey animals. The deer, wild pigs, goats, and water buffalo that the Komodo dragon hunts are themselves products of island evolution, some of them reduced somewhat in size from their mainland relatives, all of them adapted to the specific vegetation and terrain of these volcanic islands. The dragon and its prey exist in a tight evolutionary relationship, each shaped by the other, each the product of the same island pressures working through different evolutionary pathways. Perhaps the most remarkable recent discovery about Komodo dragon reproduction is their capacity for parthenogenesis, the ability of females to produce offspring without fertilization by a male. This was confirmed definitively in 2006 when two female Komodo dragons in captivity at British zoos produced fertile eggs despite having been housed without males. The hatchlings were all male, which is consistent with the genetics of sex determination in monitor lizards, where females carry two of the same sex chromosome and males carry two different ones. A female reproducing alone can only produce offspring carrying her own chromosomes, which results in either a specific combination that produces males or non-viable embryos, in the Komodo case, viable males. This is not merely a curiosity of captive animals. It has real ecological and evolutionary significance for island populations. If a single female Komodo dragon were to colonize a new island, swept there by a storm perhaps, or arriving on driftwood, she would be capable of founding a new population entirely on her own. She could produce male offspring without a mate, and those male offspring could subsequently mate with her or with each other's offspring, establishing a genetically viable population from a single founding individual. This is island colonization taken to its logical extreme, a species so well adapted to the challenges of isolation that it has evolved the capacity to overcome the most fundamental obstacle of island founding, the need for a reproducing pair. The combination of traits in the Komodo dragon, its size, its hunting efficiency, its metabolic flexibility, its capacity for parthenogenesis, represents a genuinely extraordinary evolutionary achievement. And that achievement is only comprehensible in the context of island life. The Komodo dragon is not just a large lizard. It is the product of millions of years of island-specific selection, a living argument for the creative power of isolation, a demonstration that when the rules of mainland evolution are suspended, life finds astonishing new solutions. But the Komodo dragon represents one specific evolutionary trajectory on one specific set of islands. To fully appreciate the scope and universality of the island rule, we need to look at the phenomenon from multiple angles in multiple locations across multiple taxonomic groups. And no place on Earth illustrates the island principle more dramatically, more completely, or more heartbreakingly than New Zealand. New Zealand is, in many ways, the most extraordinary natural experiment in island evolution that the planet has ever produced. It is not a small island. The combined area of the North and South Islands is comparable to the state of Colorado, and the archipelago includes numerous smaller islands. But New Zealand was isolated long enough and completely enough to develop an ecology unlike anything else on Earth. And the key to understanding what made New Zealand so extraordinary is understanding what was absent from it for most of its evolutionary history: land mammals. New Zealand separated from the supercontinent Gondwana approximately 80 million years ago, at a time when modern mammals were still in their earliest evolutionary stages. When the landmass that would become New Zealand drifted away into the southern Pacific, it took with it a collection of early Gondwanan fauna, but essentially no ancestors of the modern mammal groups that would later dominate global ecosystems. No ancestors of modern rodents, carnivores, ungulates, or primates made it onto that departing landmass. New Zealand was set adrift into isolation carrying a biological cargo that did not include the mammals that would, over the following 80 million years, come to dominate every other terrestrial ecosystem on Earth. The result was an ecosystem built on an entirely different template. Without mammals competing for food and filling ecological niches, and without large mammalian predators driving the evolution of prey species, New Zealand's ecology was shaped almost entirely by birds and reptiles. Birds, which colonized the islands repeatedly over millions of years through flight, diversified into ecological roles that on every other continent were occupied by mammals. They became the grazers, the browsers, the predators, the scavengers. They filled every niche that mammals filled elsewhere, and in doing so, they became something extraordinary. The most iconic of New Zealand's bird lineages are the moa, a group of enormous flightless birds whose fossils, and in some cases, remarkably preserved remains including feathers, skin, muscle tissue, and even stomach contents, have been found across both the North and South Islands. Moa were not a single species, but a diverse group comprising at least 9 species ranging in size from turkey-like birds to absolute giants. The largest moa, Dinornis robustus, the South Island giant moa, stood up to 3.5 meters in height and may have weighed as much as 250 kilograms, making it the tallest bird known to have ever lived. What is remarkable about the moa is not just their size, but what that size represents in the context of island evolution. Moa were large because there was nothing on New Zealand to prevent them from being large. They were herbivores in an environment with no large mammalian herbivores competing with them and no large mammalian predators hunting them. They could grow as large as the food supply allowed, and New Zealand's lush, diverse vegetation, forests, shrublands, grasslands could support creatures of considerable size. The moa diversified into a suite of ecological roles. Different species occupied different habitats, consumed different types of vegetation, and ranged across different altitudinal zones. They were in effect playing the roles of deer, cattle, and sheep, animals that did not exist in New Zealand and would not exist there until human beings brought them. The absence of mammalian predators had another profound effect on New Zealand birds that we will explore in more depth when we discuss behavioral changes. It allowed birds to lose their ability to fly. Flight is extraordinarily energetically expensive. A bird spends a significant portion of its metabolic budget maintaining the muscles, the hollow bones, the specialized respiratory system, and the feather maintenance that flight requires. If there is no predator pressure that makes flight necessary for survival, evolution, which always favors efficiency, will tend to reduce or eliminate flight over time. Flightlessness is not a disability in a predator-free environment, it is a cost saving, and New Zealand's birds embraced it enthusiastically. The moa lost not only flight but the very wings themselves. Unlike other flightless birds such as ostriches and emus, which retain small vestigial wings, the moa had essentially no wing bones at all, just tiny remnant structures buried within the shoulder girdle. They had been flightless for so long, and the selection pressure for eliminating the energy cost of wings had been so consistent that evolution had stripped them down to nothing. Imagine standing in a forest on prehistoric New Zealand and looking up at a 3.5-meter bird that had no wings whatsoever. Not tiny wings, not vestigial wings, no wings. That is what natural selection produces when given 80 million years of island isolation and no reason to fly. But if the moa were the most impressive of New Zealand's giant birds, they were not the only remarkable avian evolutionary product of this extraordinary island. The kiwi, now a national symbol of New Zealand, so deeply ingrained in the culture that people from the country call them same, is in many ways even more evolutionarily interesting than the moa, precisely because it is still alive. Kiwi are small, nocturnal, flightless birds with long, curved bills equipped with nostrils at the tip, making them the only birds in the world with nostrils at the end of their bill rather than at the base. They cannot fly. They have vestigial wings completely hidden beneath their body feathers. They have a sense of smell that is extraordinary for a bird and more reminiscent of a mammal. They forage for invertebrates at night, probing their long bills into the soil to find earthworms and grubs, using smell rather than sight. They lay eggs that are, relative to body size, the largest of any bird on Earth. A kiwi egg can constitute up to 20% of the female's body weight. The kiwi is not following the pattern of the moa. It has not become a giant. It has instead evolved into an ecological niche that, on any other continent, would be occupied by a small mammal. The kiwi is, in behavioral and ecological terms, essentially a ground-dwelling mammal in a bird's body. Its flightlessness, its nocturnality, its reliance on smell over sight, its egg size, all of these traits make sense when you understand that the kiwi evolved in an environment where small mammals did not exist and the nocturnal, soil-foraging ecological niche was empty. The kiwi filled it. It became the thing that was missing. And it did so thoroughly that it developed traits more reminiscent of a hedgehog or a shrew than of any other bird. The kakapo, whose scientific name is Strigops habreptilis, takes this mammal mimicry even further. The kakapo is a parrot, a large, flightless parrot. It is the world's heaviest parrot, with adults reaching up to 4 kilograms. It is also the only flightless parrot in the world and the only parrot that is nocturnal. Its face is lined with distinctive, bristly feathers that function as sensory organs, exactly the kind of facial whisker arrangement you would expect to find on a nocturnal, ground-dwelling mammal. The kakapo has a musty, sweet smell, again unusual for a bird, but consistent with an animal that communicates in ways more typical of mammals. Males gather in specific display areas called leks during breeding season and boom out low-frequency calls that can carry several kilometers through forest, an unusual breeding system that, again, has more parallels in mammalian ecology than in bird ecology. The kakapo evolved in the absence of mammalian predators and mammalian competitors. It has a very slow reproductive rate, lives for an extraordinarily long time, up to 90 years, and is oriented ecologically toward the kind of slow, low-intensity lifestyle that works in a predator-free environment where quality of life and energy efficiency matter more than rapid reproduction and vigilant escape behavior. It is, in short, a bird that has evolved to fill a mammal's ecological role so thoroughly that it has independently acquired many of the traits that define mammals without sharing any mammalian ancestry. This phenomenon, unrelated species in different places, independently evolving similar traits in response to similar ecological pressures, is called convergent evolution, and islands are its most spectacular stage. When the same ecological niche exists in different isolated environments, natural selection tends to produce similar solutions, regardless of what the starting material is. This is one of the most profound insights that island biology offers to our understanding of evolution, that natural selection is, in a sense, repeatable. Given the same pressures, it tends to produce the same answers. Evolution is not random. It is not chaotic. It follows a logic. And that logic becomes clearest, most readable, most undeniable, on islands where the pressures are stark and the outcomes dramatic. The kakapo's story does not end in prehistoric New Zealand, of course. It has a modern dimension that we will return to when we discuss conservation. But for now, let us remain in the deep time of island evolutionary history and look at another set of islands that offers perhaps the clearest, most celebrated evidence of convergent evolution and the island rule. The Galapagos Archipelago. The Galapagos Islands sit in the Pacific Ocean roughly 1,000 kilometers west of the South American coast. They are volcanic in origin, relatively young in geological terms. The oldest emerged from the sea only about 5 million years ago, and they have been populated entirely by species that managed to cross 1,000 kilometers of open ocean. There is no connection to the mainland, no land bridge, no shallow sea passage that organisms could walk or swim across during periods of low sea levels. To reach the Galapagos, you had to fly or ride ocean currents on floating vegetation, or drift on warm Pacific swells over days or weeks of open water. The difficulty of crossing means that the Galapagos received only a small, random sample of South American biodiversity, a few species of finch, a few reptile lineages, some plants and insects. But what happened to those few colonists over millions of years of island isolation is the stuff of evolutionary legend. The Galapagos tortoises are perhaps the most iconic example of island evolution in the entire history of biology. These extraordinary reptiles, some of the longest-lived vertebrates on Earth, with individuals documented to live more than 150 years, are the largest living tortoises in the world. Adults can weigh more than 250 kilograms and measure over a meter in shell length. They are slow, patient, monumental creatures that seem to exist in geological time rather than biological time, as though they belong to the same temporality as the volcanic rock they walk across. The giant tortoises of the Galapagos represent a case of island gigantism that closely parallels the pattern we see in other reptilian island giants. Small tortoise ancestors, possibly similar to the small South American tortoises that still exist today, colonized the Galapagos and found themselves in an environment with no large predators and, depending on the island, abundant vegetation. Released from the predator pressure that constrained mainland tortoise size, they grew and grew. And over millions of years of isolation on these volcanic islands, they became the gentle giants that now serve as one of the most recognizable symbols of evolutionary science. But what makes the Galapagos tortoises especially instructive from an evolutionary perspective is not just their size, it is the variation among tortoise populations on different islands. Distinct tortoise populations on different islands within the Galapagos archipelago have evolved different shell shapes, different neck lengths, different behavioral tendencies, all in response to the specific ecological conditions of their particular islands. On islands with abundant low-growing vegetation, tortoises tend to have dome-shaped shells and short necks. The domed shell is stronger, provides better protection, and works well for an animal that browses close to the ground. On islands where vegetation is sparse at ground level but more abundant at higher reaches, on cactus pads, for instance, tortoises have evolved a distinctive saddleback shell shape, where the front of the shell curves upward, allowing the tortoise to extend its long neck dramatically to reach food sources high above the ground. These shell-shape differences are not superficial. They represent significant anatomical reorganization, driven entirely by the specific resource availability on specific islands. The same founding lineage, subjected to different island pressures on different Galapagos islands, produced different evolutionary outcomes. This is convergent and divergent evolution working simultaneously. Convergence in overall body size increase. All the Galapagos tortoise populations share island gigantism, but divergence in specific adaptations to specific islands. Charles Darwin famously visited the Galapagos in 1835 during the voyage of the Beagle, and his observations of the island's extraordinary fauna, particularly the finches and the tortoises, contributed importantly to his eventual development of the theory of evolution by natural selection. Darwin noticed that tortoises on different islands looked different, and that local inhabitants could tell which island a tortoise came from just by looking at its shell. He noticed that the finches, which we now know to be a diverse radiation of birds descended from a single South American ancestor, had diversified into an extraordinary variety of beak shapes and sizes, each apparently well suited to a different food source. Large, heavy beaks for cracking seeds, long, probing beaks for reaching insects in wood, sharp, cutting beaks for tearing flesh. These were not different species that had arrived separately from the mainland. They were descendants of a single ancestral finch that had diversified over millions of years of island evolution into a remarkable array of ecological specialists. The Galapagos finches, or Darwin's finches as they are often called, are a textbook example of what biologists call adaptive radiation, the rapid diversification of a single ancestral lineage into multiple ecologically distinct forms, driven by the availability of unfilled ecological niches. On the mainland, each of the food sources that the different Galapagos finch species exploit would already have a specialist exploiting it. There would be dedicated seed crackers, insect hunters, and so on, already established and competing. On the Galapagos, the niches were empty. The ancestral finch lineage that reached the islands found a smorgasbord of unexploited ecological opportunities, and natural selection, the relentless process of differential reproductive success, drove the lineage to explore and fill them. Adaptive radiation is one of the most spectacular and common evolutionary phenomena on islands. The Hawaiian honeycreepers, another radiation of birds from a single founding lineage, show a similar pattern of beak diversification. The Annelise lizards of the Caribbean have radiated on individual islands into specialists for different microhabitats, different species for treetops, for mid-level branches, for ground surfaces, for grass stems. Each island's Annelise radiation has independently produced similar ecological specialists, again, convergence, with the same ecological niches being filled by related but distinct species on each island. The lesson is consistent and striking. When a group of organisms arrives on an island with empty ecological niches, it diversifies, it fills the vacancies, it explores the available evolutionary space. And because the same ecological vacancies exist on different islands, different founding lineages independently produce similar solutions. Islands are machines for generating both evolutionary diversity and evolutionary convergence, simultaneously, paradoxically, magnificently. But ecological opportunity is only one half of the island evolutionary equation. The other half, equally powerful, and in some ways more immediately legible to human observers, is the behavioral transformation that islands impose on their inhabitants. And to understand this, we need to think about one of the most important behavioral traits that island isolation tends to erode: fear. On the mainland, the animals that survive are the ones that are appropriately afraid of the right things. A deer that does not respond to the scent of a predator, or a bird that does not take flight at the approach of a hawk, or a lizard that does not dash for cover when a shadow passes overhead, these animals do not survive. They are eaten. Their genes, including whatever genetic variants made them insufficiently fearful, are removed from the population. Over generations, this relentless predator-driven selection produces animals that are vigilant, reactive, and appropriately afraid. Fear in a predator-rich environment is not a neurotic response. It is an adaptive one. It is a survival mechanism honed over millions of years of being hunted. Now take those animals or their descendants to an island without predators. The deer that wastes metabolic resources constantly scanning for threats that do not exist is at a disadvantage compared to the deer that is relaxed and devotes those resources to feeding and reproduction. The bird that wastes time and energy fleeing from shadows is less reproductively successful than the bird that continues foraging undisturbed. The lizard that retreats into crevices every time something moves overhead is less efficient at thermoregulation than the one that stays in the sun. Fear is no longer adaptive. It becomes a cost without a benefit, and evolution does not maintain costs without benefits. The result, observed consistently on predator-free islands around the world, is that island animals lose the behavioral responses associated with predator avoidance. They become fearless, or more precisely, they lose the evolved behavioral responses that serve as defense mechanisms against predators they have never encountered and do not encounter. And this makes them extraordinarily vulnerable, perhaps the most important and heartbreaking insight of island ecology when new predators eventually arrive. We will explore this vulnerability in detail as our story progresses. But first, let us look at one of the most extraordinary products of island isolation, a case that synthesizes virtually everything we have discussed so far: the dwarfism, the behavioral changes, the evolutionary flexibility of island life, into one of the most remarkable and debated discoveries in the history of paleoanthropology. Let us travel to the island of Flores, Indonesia, and meet our own tiny relative. In 2003, a team of Australian and Indonesian researchers excavating a cave called Liang Bua on the island of Flores made a discovery that would reshape our understanding of human evolutionary history and ignite one of the most heated scientific debates of the early 21st century. In the sediments of this cave, they found bones, hominin bones. But not the bones of modern humans or of the large-bodied Homo erectus that they knew had reached Asia hundreds of thousands of years earlier. These were the bones of something different, something small. The individual that became known as LB1 and whose discovery was announced to the world in 2004 was an adult female, probably. The pelvis and tooth wear suggested an adult, but she stood only about 1 meter tall. Her brain volume, estimated from the skull, was approximately 400 cubic centimeters, similar to a chimpanzee, and roughly 1/3 the volume of a modern human brain. Her wrist bones, her shoulder morphology, and certain features of her feet were distinctly archaic, resembling the anatomy of much more ancient hominins than Homo erectus or modern humans. And yet she was found in deposits dated to around 60,000 years ago, long after Homo sapiens had spread across much of the world. The scientific team named the new species Homo floresiensis, and they proposed something that seemed almost impossible: that the island of Flores had harbored a population of hominins that had evolved dwarfism through the island rule, shrinking from large-bodied ancestors to the diminutive proportions of LB1 and the other individuals subsequently found in the cave. They were not humans, and they were not Australopithecines, and they did not fit neatly into any existing category of the human family tree. They were something new, something the evolutionary history of our own lineage had produced under the extraordinary pressures of island isolation. The discovery was greeted with a mixture of awe and skepticism that has rarely been equaled in the history of paleoanthropology. Some researchers immediately accepted the proposed new species interpretation, excited by the revolutionary implications for human evolution. Others argued that the small-bodied individuals from Liang Bua were simply modern humans with a pathological condition, microcephaly being the most commonly proposed condition that caused their small brain size. This debate was fierce, sometimes personal, and played out in the pages of the world's most prestigious scientific journals over the course of more than a decade. The weight of evidence, as it has accumulated over the years since the initial discovery, has increasingly supported the new species interpretation. Multiple individuals have now been found, ruling out individual pathology as an explanation for all of them. The anatomy of Homo floresiensis is consistently different from modern humans in ways that cannot be explained by a single pathological condition affecting only the skull. The wrist bones, the shoulder structure, the foot morphology, these anatomical features do not match any known pathological condition in modern humans, but they do fit with the anatomy of older, more primitive hominins. Furthermore, the island context makes perfect evolutionary sense. Flores was indeed an island during the Pleistocene, separated from mainland Asia and other large islands by deep water channels that would not have been fully crossable on foot even during periods of low sea level. A founding population of Homo erectus, the most likely ancestor of Homo floresiensis, given the erectus fossils known from nearby Java, reaching Flores would have been subject to precisely the island pressures we have been discussing throughout this documentary. Limited resources on a small island would have created consistent selection pressure for reduced body size. The same logic that shrank Mediterranean elephants and produced the dwarf stegodons of Flores itself would have worked on hominins, and the numbers support this conclusion. Homo erectus on the Asian mainland stood about 1.5 to 1.8 meters tall and had a brain volume of roughly 900 to 1,000 cubic centimeters. The journey from those proportions to the meter-tall, 400 cubic centimeter brain Homo floresiensis is dramatic, but it is not, in the context of island dwarfism in megafauna, unprecedented. The dwarf elephants of the Mediterranean reduced to 4% of their mainland ancestors' body mass. The reduction in Homo floresiensis, while extreme, is within the range of what island dwarfism is known to produce in other mammals. What makes Homo floresiensis particularly extraordinary, what gives it a place in this documentary that goes beyond mere curiosity, is what appears to have been preserved along with the reduced body size. Despite their small brains and diminutive bodies, Homo floresiensis left behind evidence of sophisticated behavior. Stone tools were found in association with the fossils, not simple, crude tools, but a variety of tool types showing evidence of careful manufacture. There is evidence that the Flores hominins hunted stegodon, the dwarf stegodon elephant that we mentioned earlier, as well as giant rats and Komodo dragons. Fire may have been used. The complexity of the behavioral evidence associated with Homo floresiensis suggests that their small brains were not functioning simply as scaled-down versions of erectus brains, but were organized to produce behaviorally sophisticated output despite their reduced volume. This raises one of the most fascinating and unresolved questions in paleoanthropology. How did Homo floresiensis maintain complex cognitive and behavioral capacities in a brain less than half the volume of its probable ancestor? Did the brain reorganize internally as it shrank, becoming more efficient but differently structured? Did it lose some cognitive capacities while maintaining others? Did cultural transmission, the passing of learned skills and knowledge from generation to generation, allow behavioral complexity to persist even as the neural substrate that produced it became physically smaller? We do not yet know. The questions are profound, and they go to the heart of what it means to be cognitively complex, what the relationship between brain size and intelligence actually is, and whether intelligence is as much a cultural and behavioral phenomenon as it is a purely neurological one. Homo floresiensis forces us to question assumptions we barely knew we were making. It is an island species in the deepest sense, not just a creature shaped by island isolation, but a creature whose very existence challenges the boundaries of how we understand our own lineage. The most recent dating evidence from Liang Bua suggests that Homo floresiensis survived on Flores until at least 50,000 years ago, and possibly more recently. This means they were alive on Flores while Homo sapiens was spreading across Asia and Australasia, potentially the same time period when modern humans were crossing the waters around Flores on their way to populate Australia. Whether Homo sapiens and Homo floresiensis ever encountered each other is not known. But the timing raises the tantalizing possibility that two different human species, one the tiny island dwarf product of hundreds of thousands of years of Flores isolation, the other the large-brained, technologically sophisticated modern human that had originated in Africa and was now spreading across the world, may have looked at each other across a forest clearing or a beach on the island of Flores. What that encounter, if it happened, was like is something we can only imagine. The island of Flores, it is worth pausing to note, is an extraordinary place in evolutionary terms. In one relatively small island, we have the convergence of dwarf stegodons, giant Komodo dragons, and dwarf hominins, three different evolutionary trajectories following the same island logic in three different lineages. Large animals getting small, small animals getting large, intelligent animals getting smaller but retaining complexity. Flores is a microcosm of everything the island rule can do, compressed into a few thousand square kilometers of volcanic Indonesian landscape. But we have been focused, thus far, primarily on the physical dimensions of island evolution, the changes in body size, the adaptive radiations, the convergent solutions to shared ecological problems. We have touched on behavioral changes, but have not yet given them the full attention they deserve. Because the behavioral transformations that islands produce in their inhabitants are in some ways even more striking than the physical ones, and they carry some of the most urgent implications for the modern conservation challenges we will explore in the second part of this documentary. Let us return to New Zealand for a moment, because New Zealand's birds provide some of the clearest and most extensively studied examples of island-induced behavioral change. We have already noted that the absence of mammalian predators on New Zealand allowed many of its birds to become flightless, a physiological change driven by selection against the metabolic cost of maintaining flight in an environment where flight was not necessary for predator avoidance. But flightlessness is, in a sense, a behavioral consequence as much as a physical one. A bird that is flightless is a bird that has stopped using a behavior, flight, that was once essential for survival. And a bird that does not use a behavior does not need the neurological infrastructure, the muscular investment, or the physiological overhead to support it. Over evolutionary time, the physical capacity for flight atrophies when the behavioral reliance on it disappears. But the behavioral changes in New Zealand's birds go far beyond merely losing flight. The birds of pre-human New Zealand had lost, or substantially reduced, a whole suite of behaviors associated with predator avoidance and vigilance. They were, by mainland standards, extraordinarily bold, slow-moving, and approachable. Early European explorers in New Zealand and later ornithologists studying fossil and subfossil populations documented the remarkable fearlessness of native New Zealand birds. They would walk up to these birds without the animals showing any alarm response. Birds that should, by any mainland standard, have burst into flight at the approach of a large mammal would instead stand their ground or approach out of curiosity. This was not stupidity. It was rational behavior given the evolutionary environment these birds had existed in for millions of years. There was nothing to be afraid of. No large predators had ever existed in New Zealand before humans arrived. The behavioral responses to predator approach, the alarm calls, the freezing, the explosive escape flights, were metabolically and reproductively costly to maintain, and since they had never been required, natural selection had gradually reduced them. The birds had evolved confidence in a world that was genuinely safe for them. Then humans arrived. The Polynesian ancestors of the modern Maori people reached New Zealand approximately 700 to 800 years ago, and later European settlers arrived in the 19th century. And the world that had been safe for 65 million years of New Zealand bird evolution became, almost overnight in geological terms, catastrophically dangerous. Humans hunted. Humans brought rats which had colonized Polynesian voyaging canoes. Humans brought dogs. Humans brought, eventually, stoats, weasels, ferrets, and other predators. And the birds of New Zealand, their behavioral defenses eroded by millions of years of predator-free evolution, had absolutely no response to any of this. The moa were extinct within a few centuries of human arrival, almost certainly due to hunting. A creature that stands 3.5 meters tall and has no fear of approaching bipedal primates is not difficult to hunt. The giant moa was possibly the most hunted large animal in the history of human colonization, not because it was particularly numerous, but because it was utterly defenseless against a predator it had never evolved to recognize. The kakapo, with its slow reproduction, its flightlessness, and its ineffective defense responses, was similarly decimated. Introduced stoats are among the most devastating predators for kakapo, a small, quick mustelid carnivore that can pursue the flightless parrot into any crevice or forest corner. The kakapo has a stress response to predators. It freezes and relies on its cryptic coloration, a response that works reasonably well against visually oriented predators, but is entirely ineffective against a stoat hunting by smell. The story of New Zealand's birds is, in this dimension, a tragedy. The same evolutionary forces that made them extraordinary, the island rule working over millions of years to produce creatures perfectly suited to their predator-free environment, made them catastrophically vulnerable to the changes that human arrival brought. Their fearlessness, their flightlessness, their slow reproduction, their specialization to a specific and now disrupted ecology, all of these traits that were adaptive in the world they evolved in became, almost overnight, lethal liabilities in the world that arrived with us. But the story of New Zealand's birds is not only a tragedy. It is also, increasingly, a story of conservation science fighting back with growing sophistication. We will return to that in detail. But for now, let us move from the ancient to the startlingly contemporary, from the fossils of dwarf elephants and giant moa to an island where the island rule is visible in real time, happening right now, with measurable results that scientists have been documenting over decades. Go Island sits in the South Atlantic Ocean, roughly halfway between southern Africa and the tip of South America. It is a remote, wind-scoured place, a volcanic island of about 91 square kilometers that rises steeply from the ocean's surface to a plateau of alpine grassland and heath. It is, by any measure, one of the most isolated inhabited places on Earth. The nearest land is Tristan da Cunha, the most remote permanently inhabited island in the world, 250 kilometers away. Before humans discovered Go Island in the 15th century, it was entirely free of land mammals. Its ecosystem was dominated by seabirds, millions of them, nesting in enormous colonies on the island's slopes and cliffs. Albatrosses, petrels, storm petrels, and shearwaters in numbers that astonished early mariners. House mice, Mus musculus, arrived on Go Island sometime in the 19th century, almost certainly on sealing or whaling vessels that made stops at the island. Mice are extraordinarily successful colonizers, one of the world's most prolific and adaptable mammal species. The Go Island population established itself in the usual house mouse fashion, feeding initially on seeds, invertebrates, and whatever food scraps were available near the small research station that was eventually established on the island. But then something happened that was not usual house mouse fashion at all. Something that should not, by any reasonable expectation of house mouse behavior, have happened. Something that shocked the scientists who first documented it and continues to fascinate evolutionary biologists today. The mice began attacking and eating seabird chicks alive. This bears repeating because it is such an inherently improbable thing. House mice, tiny, typically prey animals, began attacking Tristan albatross chicks. Tristan albatross chicks are among the largest seabird chicks in the world. At the point when mice begin attacking them, they can weigh as much as 4 kilograms, roughly twice the body weight of the mice doing the attacking. The attacks, documented by researchers through remote cameras and field observation, involve multiple mice working together, gnawing through the chick's skin while the bird is alive and unable to defend itself. The chick, evolutionarily naive to any threat from small mammals, because small mammals had never existed on Go Island before, does not flee. Does not effectively fight back. It sits in its nest and is eaten alive. This behavior has had catastrophic consequences for the albatross colonies on Go Island. The Tristan albatross, already a species of conservation concern due to longline fishing mortality at sea, has seen its chick survival on Go Island plummet. Studies have estimated that mice kill more than 2 million seabird chicks and eggs on Go Island every year. This is not a trivial figure. The island hosts globally significant populations of several seabird species, and mouse predation is driving some of these populations toward functional extinction. But from an evolutionary perspective, what is happening on Go Island is not merely tragic. It is extraordinary. The Go Island mice have evolved in real time, within perhaps 200 years of house mouse history on the island, to be significantly larger than house mice anywhere else in the world. Studies have confirmed that Go Island mice are roughly 50% larger in body mass than mainland house mice. They have shifted from the standard house mouse diet to a diet that includes substantial amounts of animal protein, seabird flesh, and eggs, and they have developed cooperative hunting behaviors that are not observed in any other house mouse population. This is the island rule in action, not in the fossil record, not in the deep past. Right now in the 21st century, in a population of animals that colonized an island less than 2 centuries ago, the mice have responded to the island environment, abundant food, no predators, evolutionary naivety of prey, with precisely the size increase and behavioral shift that the island rule predicts. They are island animals now. They are Go Island mice, not merely house mice on an island. Natural selection has already had enough time to begin reshaping them. The speed of this change is what is most remarkable. 200 years is perhaps 50 to 100 mouse generations, depending on reproductive rate. 50 to 100 generations is a blink of an eye in evolutionary time. And yet already, significant and measurable changes in body size and behavior have accumulated in the Go Island population. This tells us something crucial about the island rule that can sometimes get lost when we focus on ancient fossils in deep time. It can work fast. When the selection pressure is strong, as it is on islands, where ecological conditions differ dramatically from the mainland, evolution does not have to wait for geological ages to produce observable change. Go Island mice are also teaching us something important about behavioral evolution. The cooperative hunting behavior shown by these mice, multiple individuals working together to overwhelm a prey animal much larger than any single mouse could tackle, is a learned tradition as much as it is an evolved behavior. Young mice on Go Island learn from older mice how to attack seabird chicks. This cultural transmission of behavior means that the predatory behavior can spread and be refined much faster than genetic evolution alone would allow. The island rule is not just genetic adaptation. It is behavioral adaptation, cultural adaptation, and genetic adaptation all working together, all driving toward the same outcome, an animal increasingly well suited to the specific conditions of its island home. The story of Go Island mice is, like the story of New Zealand's birds, a story with conservation dimensions that we will address in the second part of this documentary. But it is also a story about time scales, about the reminder that evolution is not something that happened in the distant past and is now complete. Evolution is happening right now. Islands are laboratories where we can watch it happen in real time, and the island rule is not an ancient, completed process. It is ongoing, active, and responsive to the conditions of the present moment. This realization that island evolution is not just historical but contemporary is one of the most important shifts in perspective that the study of island biology offers. When we look at a Komodo dragon or a giant Galapagos tortoise or the fossils of a dwarf elephant, it is tempting to see them as relics of evolutionary processes that have run their course. The Komodo dragon evolved, the tortoise evolved, the dwarf elephant evolved. And now they simply exist, static and complete, waiting for us to observe them. But that is not how evolution works. Every island population on Earth today is still evolving, still responding to the pressures of its environment, still changing. The kakapo of New Zealand is not a finished product. It is a species in the middle of its evolutionary story, currently at a crisis point that will determine whether it continues that story or ends it. The tortoises of the Galapagos are not done adapting. Their populations on different islands continue to diverge, continue to respond to the specific conditions of their habitats. The mice of Go Island are in the early chapters of what, if they had the time, would become a far more dramatic evolutionary transformation. And this is why island biology matters for the present and the future as much as it matters for the past. The principles that produce island dwarfism and gigantism, that drive convergent evolution and adaptive radiation, that erode fearfulness and generate behavioral novelty, these principles do not stop operating because we have discovered them and given them names. They continue. They respond to whatever pressures the environment provides. And in the modern world, the pressures on island species are more intense, more sudden, and more catastrophically disruptive than anything the island rule has had to deal with in its long evolutionary history. Because the island rule was designed, if we can use that word loosely, metaphorically, for a process that has no designer, for a world where change happens slowly, where new predators arrive rarely, over geological time scales, in ones and twos that the ecosystem can absorb and adapt to. Where new competitors establish themselves gradually, giving existing species time to respond evolutionarily. Where habitat changes unfold over thousands or millions of years, leaving enough time for natural selection to track the shifting conditions and adjust. The island rule was not designed for us. It was not designed for the speed at which modern humans arrive on islands, or the number of invasive species we carry with us, or the rate at which we transform habitats. It was not designed for a world in which a species that has spent millions of years evolving in the absence of mammalian predators suddenly has to contend with rats and cats and stoats and snakes introduced in the space of a few centuries. The extraordinary specialization that island evolution produces, the precise tuning to a specific environment, the behavioral naivety toward predators that never existed, the slow reproductive rates adequate for a stable, low-risk world, all of these become fatal vulnerabilities in the face of rapid, human-driven change. We are not yet at the conservation part of this documentary. That will come. But it is important to hold this awareness as we continue exploring the science of the island rule, to understand that we are not talking about something abstract or purely academic. We are talking about creatures that exist right now, on islands right now, that are the products of evolutionary histories so extraordinary they exceed anything human imagination could invent, and that are under threats so severe that their continued existence cannot be taken for granted. Komodo dragon faces habitat pressure. The kakapo teeters at the edge of extinction. The albatrosses of Go Island are being eaten by mice. The giant tortoises of the Galapagos, the kiwi of New Zealand, the Aldabra tortoise, the dwarf hippopotamus of Madagascar, these are not just evolutionary curiosities. They are the living products of millions of years of island evolutionary history, and they are fragile in ways that their extraordinary evolutionary heritage does nothing to protect against. But let us not end this section of our exploration on a note of pure elegy. Let us instead take stock of what we have learned so far and look ahead to the next phase of our journey into island evolution. We have established the basic mechanics of the island rule. Large animals shrink in response to resource limitation on islands. Small animals grow in the absence of predator pressure. We have met the dwarf elephants of the Mediterranean, the giant tortoises of the Galapagos, the extraordinary birds of New Zealand, the remarkable Komodo dragon. We have visited Flores and encountered the startling, brain-bending case of Homo floresiensis. We have seen the island rule working in real time on Go Island through the behavioral and physical transformation of house mice. We have begun to understand the behavioral changes that island isolation induces, particularly the erosion of predator avoidance responses, and we have glimpsed how these behavioral changes, so adaptive in the island environments that produced them, become catastrophic liabilities when human-driven change arrives. In the next section of our exploration, we will go deeper into the mechanisms of convergent evolution, exploring how unrelated species on different islands around the world have arrived at remarkably similar solutions to similar ecological problems. We will look at the genetics underlying island adaptations, examining what scientists are learning at the molecular level about how island evolution actually works. We will explore more of the spectacular examples of island life that populate the world's archipelagos and remote landmasses. And we will begin to look seriously at the intersection of island evolutionary science and the urgent conservation challenges of the modern world, how understanding the island rule is helping scientists and conservationists protect and restore island ecosystems that are among the most irreplaceable and most threatened in the natural world. Because the island rule, ultimately, is not just a fascinating academic subject. It is a window into the deepest principles of how life works, how it adapts, how it transforms itself in response to the conditions it encounters. Islands are nature's laboratories, isolated, controlled, legible in ways that the vast complexity of continental ecosystems can never quite be. They allow us to see the logic of evolution with unusual clarity. They allow us to test our understanding of natural selection, of adaptation, of the relationship between genes and bodies and behavior and environment, in ways that mainland ecology cannot easily provide. And as we face a future in which human activity is creating, in effect, new islands everywhere, isolated patches of habitat surrounded by human-modified landscapes, the principles of island ecology are becoming relevant to conservation biology far beyond the literal shores of actual islands. The fragmented forests of tropical regions, the isolated wetlands surrounded by agricultural land, the mountain ranges standing alone in a sea of developed terrain, all of these are becoming, in an ecological and evolutionary sense, islands. The same principles that drive insular dwarfism and behavioral naivety and adaptive radiation on geographic islands will increasingly shape the evolution of species confined to habitat islands in a human-dominated world. Understanding the island rule, then, is not merely an exercise in appreciating the biological wonders of remote and exotic places. It is an investment in the intellectual tools we will need to navigate the biological challenges of the coming centuries. It is preparation for a world in which isolation, not the gentle, gradual isolation of geological change, but the rapid, human-driven isolation of habitat fragmentation, is becoming a dominant evolutionary force across the entire planet. We are all, in a sense, living on islands now. The question is whether we will learn the lessons that those islands have to teach us before it is too late. Let us continue the journey. One of the most powerful illustrations of convergent island evolution comes not from any single archipelago, but from comparing isolated islands across different ocean basins and different regions of the world. When you do this comparison, when you stand back and look at the pattern of island evolution globally rather than island by island, a striking regularity emerges. The same ecological niches, when empty on isolated islands, tend to be filled by whatever lineage happens to be available, and the solutions, regardless of which lineage fills them, tend to look remarkably similar. Consider the ecological role of the large ground-dwelling omnivore or generalist carnivore. On continental landmasses, this role is typically filled by mammals, bears, large raccoon relatives, civets, various mustelid species. But on islands where mammals were absent or underrepresented, other lineages have stepped in to fill comparable roles, and in doing so, have evolved traits that convergently resemble the mammals they functionally replace. The Komodo dragon's ecological role as an apex predator and scavenger on its Indonesian islands is filled, on the Galapagos, by nothing quite as dramatic, but the marine iguana and the land iguana of the Galapagos represent a different kind of convergence. Marine iguanas, Amblyrhynchus cristatus, are the only marine lizards in the world. They have evolved in the specific context of the Galapagos Islands, where food is scarce on the volcanic land surfaces but abundant in the surrounding ocean in the form of algae. Marine iguanas dive into the cold Pacific waters, holding their breath for up to 30 minutes, and scrape algae off submerged rocks with their specialized tricuspid teeth. They are reptiles that have convergently evolved a marine foraging lifestyle, a lifestyle typically associated with birds and marine mammals, not lizards. The convergence here is not that the marine iguana resembles a marine mammal physically. It is that the ecological problem, accessing a food source in the ocean, has produced a similar solution. An air-breathing vertebrate that can tolerate cold water submersion, regulate its core temperature through behavioral thermoregulation after diving, and efficiently process a primarily algae-based diet. Different anatomies, different physiologies, different evolutionary histories, but similar ecological function, driven by similar island pressures. The same principle plays out across the world's islands in example after example. Madagascar, one of the largest and most evolutionarily isolated islands on Earth, separated from Africa roughly 88 million years ago and from India about 67 million years ago. Like New Zealand, it has developed a unique fauna that reflects its long isolation from mammalian evolutionary trends on the nearby continent. Madagascar's most famous evolutionary products are the lemurs, a diverse radiation of primates that filled, in the absence of monkey and ape competition, an extraordinary range of ecological niches. There are lemur species that forage like squirrels, lemur species that behave like baboons, lemur species that are nocturnal and mouse-sized, and lemur species that were, before human arrival, gorilla-sized. The giant subfossil lemurs of Madagascar, species like Archaeoindrus fontenantii, which weighed perhaps 200 kilograms, or the koala lemur megaladapes, are gone now, driven extinct by human activity, but their fossils document an adaptive radiation as spectacular as anything produced on any island. Madagascar also produced the tenrecs, a remarkable radiation of small mammals that, in the absence of most other small mammal lineages, diversified to fill the niches of shrews, hedgehogs, otters, and various other insectivores. There are tenrec species that look and behave like moles, tenrec species that look and behave like hedgehogs, tenrec species that look and behave like otters. They are all tenrecs, all part of a single Madagascar lineage, but they have convergently evolved to resemble unrelated mammals from other parts of the world. The ecological pressures of Madagascar's specific environments, operating on the available tenrec lineage, have independently produced solutions that parallel the solutions that different evolutionary lineages produced on the continental mainlands. This pattern of convergent evolution, independent lineages arriving at similar solutions to similar ecological problems, is one of the deepest signatures of natural selection's logical consistency. It tells us that evolution is not random, not merely a process of drift and accident. It has a logic, and that logic produces predictable outcomes when the conditions are comparable. Islands, because they create comparable conditions for ecological opportunity and constraint across their global distribution, produce the clearest demonstrations of this logic. The most famous systematic study of convergent island evolution is the work done on Annelise lizards across the Caribbean islands. The Caribbean is home to hundreds of Annelise species, a phenomenal diversity for a single genus, distributed across the islands of the Greater and Lesser Antilles. Each major island or island group has its own suite of Annelise species, and those species have diversified through adaptive radiation to fill different ecological roles. What makes the Annelise case particularly compelling for understanding convergent evolution is that the same suite of ecological specialists, the same functional categories, called ecomorphs, has evolved independently on multiple large Caribbean islands. On Cuba, Puerto Rico, Jamaica, and Hispaniola, researchers have identified remarkably similar sets of Annelise ecomorphs. Species specialized for the tops of tree canopies, species specialized for mid-trunk positions, species specialized for broad, low branches, species specialized for grass and twig habitats at or near the ground. Each island set of ecomorphs includes representatives of each habitat category. And the lizards in the same habitat category on different islands, though they are distinct species, descended from different founding lineages, and separated by millions of years of independent evolution, look remarkably similar to each other. The twig specialist Annelise on Cuba looks like the twig specialist Annelise on Jamaica. The canopy specialist on Puerto Rico resembles the canopy specialist on Hispaniola. Similar body size, similar limb proportions, similar color patterns, similar behavior. This is convergent evolution documented with a precision and rigor that rivals the best experimental science. The same ecological pressures, the same set of available microhabitats, the same food resources, the same predators, on different islands have independently produced the same suite of evolutionary solutions in lizard lineages that had no common ancestor particularly close in time. Natural selection, working on different lineages on different islands, has produced the same functional outcomes because the functional problems were the same. The Annelly system has been particularly well studied because these lizards breed quickly, are easy to observe, and can be studied both in their natural island environments and in experimental settings. Researchers have actually been able to watch convergent evolution happen in real time by observing Annelly's populations that were deliberately or accidentally introduced to new islands or new microhabitats. Within years to decades, again, an extraordinarily short evolutionary timeframe, these populations begin shifting toward the ecomorph appropriate to their new environment. Twig specialists introduced to a broad-branched environment begin evolving toward the broad-branch ecomorph. Canopy specialists introduced to a grass habitat begin evolving toward the grass ecomorph. The system is responsive, predictable, and fast. This responsiveness is a reminder that we should not think of the island rule as a single event, a one-time transformation of a mainland species into an island form. Island evolution is ongoing, responsive, and dynamic. Island populations are not simply the products of an evolutionary event that happened in the past. They are evolving systems, constantly adjusting to their conditions, capable of significant change within generations rather than millennia when the pressure is sufficient. The genetic underpinnings of this responsiveness are an active area of modern research. What happens at the level of DNA when a population undergoes island evolution? What genes change and how quickly? These questions are becoming answerable as genomic sequencing technology has become more accessible and as researchers have been able to compare the genomes of island populations and their mainland relatives with increasing precision. Some of the findings are striking. Studies comparing island and mainland populations of the same species have found that island populations often show evidence of strong selection on genes related to body size regulation. The same hormonal and developmental pathways that control growth and metabolism on the mainland are being shaped by natural selection on islands, but in consistent directions that reflect the island pressures we have been discussing. Genes associated with growth hormone signaling, insulin-like growth factor pathways, and leptin regulation, all of which influence body size in mammals, show evidence of positive selection in island dwarf populations and in island gigantized populations. Particularly interesting is the evidence for rapid genetic change in founder populations, the small groups of individuals that first colonize an island and from which the entire island population descends. Founder affects the random sampling of only a subset of the mainland population's genetic variation when a small founding group establishes itself on a new island, can themselves influence the evolutionary trajectory of the island population, regardless of selection. If the founding group happens, by chance, to include a higher frequency of individuals with certain genetic variants related to body size, behavior, or physiology, those variants will be overrepresented in the island population from the start, and selection can subsequently build on them rapidly. This interaction between random founder effects and deterministic natural selection is one of the most nuanced aspects of island evolution. It means that the evolutionary outcomes on islands are influenced both by chance, which individuals happen to survive the colonization event, and by selection, which individuals happen to survive and reproduce best in the island environment. The relative contributions of these two forces vary with the size of the founding population, the strength of the selection pressure, and the amount of time since colonization. Disentangling them in specific cases is one of the more technically demanding challenges in island evolutionary biology, but what emerges from the genomic studies consistently is a picture of evolution that is far faster, far more deterministic in its outcomes, and far more legible in its mechanisms than evolutionary science once assumed. Islands are not just evolutionary curiosities. They are evolutionary fast lanes, environments where the normal pace of change is accelerated by the intensity and specificity of the pressures involved. And the genomic revolution in biology is giving us, for the first time, the tools to read the detailed evolutionary history written in the DNA of island species, to reconstruct not just what happened, but how it happened, and sometimes even when. These genetic insights have practical as well as theoretical importance. Conservation of island species increasingly relies on genetic knowledge, understanding the population structure of threatened species, identifying distinct evolutionary units that should be prioritized for protection, assessing the genetic health of small isolated populations that may be at risk of inbreeding depression. As we move toward the second section of this documentary, where conservation takes center stage, the genetic dimension of island biology will become increasingly relevant. But we are not yet at the end of our exploration of the science. There remains one more dimension of island evolutionary biology that we need to examine before we turn to conservation, a dimension that connects the science of island evolution to some of the deepest questions in ecology and evolutionary theory, and that has implications far beyond the literal island environments where it has been most clearly studied. That dimension is coevolution, the process by which species on an island evolve in response not just to the physical environment, but to each other. On islands, where species diversity is typically lower than on mainlands and ecological relationships are simpler and more legible, coevolution can be studied with unusual clarity. And the results of island coevolution are some of the most extraordinary biological relationships in the natural world. We have already touched on this briefly in discussing the Komodo dragon and its prey species, the way that the dragon and the deer and pigs of its island habitat have shaped each other over evolutionary time. But let us look at a more detailed and more precisely studied example, the extraordinary coevolution between New Zealand's moa and the plants that had to survive being browsed by them. The moa, as the dominant large herbivores of New Zealand for millions of years, exerted enormous selective pressure on the island's plant communities. Plants that were heavily browsed by moa evolved defenses against them, and because moa evolved over the same time period and had to respond to plant defenses to survive, there was a continuous evolutionary dialogue between browser and browsed. This dialogue produced some remarkable plant morphology. Some New Zealand plant species show divaricating growth forms, a dense, tangled, small-leaved, wiry branching architecture that makes it physically difficult for a large-headed herbivore like a moa to get its mouth around the vegetation. The divaricating form is common enough in New Zealand plants and rare enough globally that researchers have proposed moa browsing as a significant evolutionary driver of this trait. Similarly, several New Zealand plant species show evidence of what biologists call defensive deceptive coloration, young leaves that are reddish or brownish rather than green, resembling dead or unpalatable material and potentially deterring browsers that are searching visually for nutritious green vegetation. Again, this trait is unusually common in New Zealand compared to similar temperate vegetation elsewhere, suggesting an evolutionary history of browsing pressure that is specific to the New Zealand context. These plant adaptations to moa browsing are a window into the hidden depths of island coevolution. They remind us that island evolution is not just about the most visible, most charismatic species, the giant tortoises and the dwarf elephants and the dragon lizards. It reaches into every corner of the ecosystem. It shapes the architecture of forests and the chemistry of leaves. It influences the timing of fruiting seasons and the structure of flowers. Every organism in an island ecosystem is part of an evolutionary web in which every thread influences every other thread, and the isolation of the island ensures that this web is a closed system. Changes in one part of the web ripple through all the others. The moa coevolution story has a poignant coda. When moa went extinct following human arrival, many of the plants that had evolved in response to moa browsing were left without the evolutionary partner they had been shaped to interact with. The divaricating growth forms, the defensive coloration, these adaptations no longer serve their original purpose because the moa that drove their evolution is gone. The plants have what biologists call evolutionary anachronisms, traits shaped by an ecological relationship that no longer exists. They are, in a sense, waiting for an animal that will never return. This concept of evolutionary anachronisms is not unique to New Zealand. It appears wherever the relationship between a plant and its evolutionary partner has been disrupted by extinction. The avocado and other large-fruited tropical plants evolved their massive fruits to attract and have their seeds dispersed by large megafauna, giant ground sloths, gomphithers, other large Pleistocene mammals that are now extinct. Those plants now produce fruits that fall to the ground and rot because the animals that would have eaten them and dispersed their seeds are gone. The plants are anachronisms, perfectly adapted to an ecological relationship that exists no more. Islands, because their simpler ecosystems make these relationships more visible, are particularly productive places to study ecological and evolutionary anachronisms. And understanding anachronisms matters for conservation because the disruption of co-evolved relationships, the removal of one partner from an evolved dialogue, can have cascading effects through an entire ecosystem. Remove the moa and you disrupt the plant community. Remove the plant community and you disrupt the birds that nest in it. Remove the birds and you lose the seed dispersers. Each disruption triggers others, and the simplified nature of island ecosystems means there are fewer redundant connections to buffer against these cascades. This is why island conservation is not simply a matter of protecting individual species. It is a matter of protecting ecological relationships, the co-evolved webs of interaction that have been shaped over millions of years and that cannot be recreated or replaced on any human timescale. A tortoise removed from an island is not simply one tortoise gone. It is the loss of a grazer that shaped the plant community, a seed disperser that maintained plant diversity, a prey item that may have supported scavengers, a soil disturber that created microhabitats for invertebrates. Its absence reshapes the entire ecosystem, and the reshaping is almost never benign. We are approaching the end of the first section of this documentary. We have covered enormous ground, from the basic mechanics of the island rule to its most spectacular examples in the fossil record and the living world. We have explored dwarfism and gigantism, adaptive radiation and convergent evolution, behavioral change and coevolution. We have met dwarf elephants and giant dragons, tiny humans and enormous tortoises, fearless birds and mouse-turned predators. We have glimpsed the genetic mechanisms underlying island change and the coevolutionary webs connecting island species to each other. What we have not yet done, what we will do in the second part of this documentary, is follow these threads into the present and the future. We will look in detail at the conservation crisis facing island species and at the extraordinary science and effort being directed toward their protection. We will explore the specific case of the kakapo and the remarkable recovery program that has brought this extraordinary bird back from the brink. We will examine the restoration science being applied to island ecosystems globally, the removal of invasive species, the reintroduction of lost ecological partners, the use of genomic science to manage breeding programs for critically endangered island populations. We will also broaden our perspective to look at the ways in which the principles of island evolution are now being applied beyond literal islands, to the fragmented habitats that are increasingly the dominant context for wildlife survival in a human-dominated world. And we will ask the deepest question that island biology poses to our own species. What is our responsibility, given our extraordinary capacity to disrupt island ecosystems, to protect and restore the irreplaceable evolutionary heritage that those islands contain? Because the island rule has been operating for millions of years, it has produced some of the most extraordinary creatures that have ever lived on this planet. It has revealed deep truths about how natural selection works, how evolution shapes body and behavior and ecology, how life responds to isolation and constraint and opportunity. It is one of nature's greatest stories, told over geological time on islands scattered across every ocean. And it is a story that we, the most powerful island-disrupting species that has ever existed, have the unique ability to either extinguish or to protect. That choice, and it is a choice, is the most important question that island biology poses to the present moment. The second part of this documentary will pursue that question with all the depth and urgency it deserves. Welcome back. If you're just joining us, please make sure to subscribe to the channel and let us know in the comments where you are watching from and what time it is there. The conversation happening in the comments on this series has been extraordinary, and we read every single one. We left the first part of this documentary standing at a threshold. We had explored the deep science of the island rule, the mechanisms of dwarfism and gigantism, the behavioral transformations that island isolation produces, the extraordinary examples that the fossil record and the living world provide. We had met dwarf elephants and dragon lizards, miniature humans and fearless birds, mice that hunt albatrosses and tortoises that live for a century and a half. We had begun to understand that islands are not merely interesting places where unusual creatures happen to live. They are nature's most powerful evolutionary laboratories, isolated environments where the logic of natural selection becomes unusually clear, unusually fast, and unusually dramatic. But we also stood at the edge of a harder conversation. The same qualities that make island species so extraordinary, their precise specialization to their environments, their behavioral naivety toward predators they have never encountered, Their slow reproductive rates calibrated to stable, low-risk worlds make them catastrophically vulnerable to the kind of rapid, disruptive change that human activity brings. The story of island evolution is, in its modern dimension, increasingly a story of crisis. But it is also, in ways that are genuinely hopeful and scientifically fascinating, a story of response, of conservation science rising to meet one of the most urgent biological challenges of our time. In this second part of our exploration, we are going to go deeper into both dimensions, the crisis and the response. We are going to look at what we know about the vulnerability of island species, why that vulnerability is so profound and so consistent across different taxonomic groups and different island environments. We are going to look at the specific science of island conservation, what works, what does not work, and what the most innovative researchers and conservation practitioners are doing right now to protect the irreplaceable evolutionary heritage that the world's islands contain. We are going to return to the kakapo, the extraordinary flightless parrot of New Zealand whose story has become, in conservation biology, one of the most intensively studied and carefully managed examples of species recovery in the world. We are going to look at the broader restoration of island ecosystems and what that means not just for individual species but for the ecological relationships, the co-evolved webs of interaction that those species are embedded in. And we are going to expand our perspective to look at the ways in which island evolutionary principles are becoming relevant to conservation far beyond the literal shores of actual islands. But let us start where the crisis starts, with the moment of contact, the moment when island isolation ends and the outside world arrives. Throughout the history of life on Earth, islands have experienced colonization events, moments when new species arrive from elsewhere and join the existing ecological community. These events are, in geological terms, rare. The isolation that defines islands means that successful colonization is an improbable event, requiring either the flight capacity to cross open water, the extraordinary luck of surviving a long ocean drift, or the coincidence of brief land connections during periods of low sea level. As a result, island ecosystems are typically assembled over very long time scales, with new arrivals appearing only occasionally, and the existing community having time to adjust and absorb each new element. The island rule and the extraordinary species it produces are partly a product of this slow pace of ecological change. When a new species arrives rarely, and in small numbers, the existing community can respond evolutionarily. Prey species can gradually develop predator avoidance behaviors in response to a new predator. Competitors can shift their resource use to reduce overlap with a new competitor. The ecosystem adjusts, absorbs, adapts. This is how coevolution works. It is a dialogue, and dialogue requires time. What human beings did when we began colonizing the world's islands was to collapse this process from geological time into historical time. Instead of new species arriving one or two at a time, over thousands or millions of years, human colonization brought dozens of new species simultaneously. The rats and dogs that came on Polynesian canoes, the pigs that were intentionally introduced as food sources, the cats and stoats and ferrets that came later with European settlers, the plants that arrived in ballast water and on the boots of explorers. Instead of millennia for ecological adjustment, island ecosystems were given years or decades to respond to changes that their evolutionary histories had given them no tools to handle. The results were, and continue to be, devastating. Island species account for a wildly disproportionate share of recorded animal extinctions since 1500. While islands contain only a small fraction of the area, they have been the site of approximately 80% of recorded bird extinctions and similar proportions of mammal and reptile extinctions since detailed historical records began. The pattern is unmistakable and consistent. Islands are where extinctions happen at rates that dwarf anything occurring on the continental mainlands. This is the island rule in its darkest expression. The same isolation that produces extraordinary evolutionary specialization makes the resulting species exceptionally vulnerable to disruption. Island species are not globally distributed populations that can withstand the loss of one regional population and recover from another. They are, by definition, found only on their island. When they go entirely. There is no backup population, no reservoir of individuals elsewhere that can repopulate. Every extinction of an island species is a permanent and total loss, not just of a species, but of millions of years of evolutionary history, of a unique evolutionary experiment that will never be repeated. Consider the scale of what has already been lost. In the Pacific Ocean alone, it is estimated that the Polynesian expansion, which began roughly 3,000 years ago and reached the most remote Pacific islands roughly 1,000 years ago, caused the extinction of roughly 1,000 bird species. 1,000 species gone before any European set foot in the Pacific. Many of these were species that left no written record, no paintings, sometimes not even fossil evidence. They simply vanished into the past, known only from bones occasionally found in archaeological sites or cave deposits. We have no idea what most of them looked like in life, what colors they were, what sounds they made, how they moved through their forest habitats. They are lost as completely as it is possible to be lost. The flightless birds of the Pacific, in particular, were decimated by Polynesian colonization. Island after island had its own suite of flightless rails, flightless ducks, flightless pigeons, birds that had, following the island rule, abandoned flight in environments where flight was unnecessary for survival. When Polynesian settlers arrived with dogs and rats, these birds had no defenses. They could not fly away. They had no instinctive fear of the approaching predator. They were, from the perspective of a hunter or a dog or a rat, simply there, abundant, slow, and unafraid. Their extinction was in most cases swift by any historical standard. The same pattern played out in Madagascar and New Zealand, in the Caribbean, in every island group that human beings reached. The first wave of human colonization was typically associated with the loss of the largest, most conspicuous, most behaviorally naive island species, the moa of New Zealand. The elephant birds of Madagascar, flightless giants even larger than the moa, with Epiornis maximus estimated at up to 500 kilograms. The giant lemurs of Madagascar, the dwarf hippos of Madagascar and Cyprus and Malta, the enormous land tortoises that had colonized islands across the Indian Ocean and Mediterranean, all gone or largely gone in the immediate aftermath of human arrival. This first wave of extinction, driven primarily by hunting and the introduction of rats and dogs, was then followed in many island groups by a second wave associated with more intensive human land use and the introduction of additional invasive species. Forests were cleared for agriculture, wetlands were drained, fire was used to manage vegetation, and with European contact came new invasives: cats, stoats, ferrets, weasels, mongooses, snakes. Diseases to which island species had no resistance. Herbivores like goats and rabbits that devastated plant communities, altering the habitat structure that remaining native species depended on. Each of these waves removed more species, disrupted more ecological relationships, and left the remaining native communities more fragmented, more isolated, and more vulnerable. The pattern has continued in various forms through the modern period. Many island species that survived the first and second waves of colonization-driven extinction are now at critically low population sizes, clinging to existence in remnant habitat patches, dependent on active human intervention for their survival. And yet, the story is not simply one of unstoppable decline. In parallel with the ongoing crisis, something else has been developing over the past several decades, something that represents one of the most encouraging developments in the history of conservation biology. Island conservation has become, in many respects, the most technically sophisticated, most rigorously scientific, and most practically successful domain within the broader field of conservation biology. The reasons for this are deeply connected to the nature of islands themselves. Islands, as we have emphasized throughout this documentary, are defined by their boundaries. They are finite, they are legible. You can survey an island completely in a way that you cannot survey a continent. You can identify every invasive species present. You can, in principle, design interventions that affect the entire ecosystem rather than just a portion of it. The boundedness that makes island species so vulnerable also makes island ecosystems uniquely amenable to active conservation management. And the conservation community has learned, over decades of trial and error and increasingly rigorous science, how to exploit that amenability with growing effectiveness. The cornerstone of island conservation has become what practitioners call island restoration, the removal of invasive species that are driving native species decline, followed by the recovery of native ecological communities. This sounds straightforward, but it is technically enormously challenging, logistically complex, and has required the development of new methods and approaches that have no precedent in conservation practice. The most fundamental component of island restoration is invasive species removal, and the most consequential invasive species to remove from islands are typically those that directly kill native wildlife: rats, cats, stoats, mongooses, and their relatives. These are the animals that, more than any others, have driven island bird extinctions, the hunters of eggs and chicks and small adults, the climbers of trees that nest, raiding flightlessness cannot protect against the relentless year-round predators that island birds evolved with absolutely no capacity to recognize or evade. Removing rats is in concept simple. In practice, it is one of the most technically demanding conservation. Rats are extraordinarily intelligent, behaviorally flexible, and reproductively prolific. A single pair of rats can produce dozens of offspring in a year, and rat populations can recover from significant culling within months if any individuals survive the eradication operation. A successful island rat eradication must achieve essentially complete removal of every rat on the island. Leaving even a small residual population is effectively the same as leaving the entire population, because rats will simply breed back to carrying capacity within a few years. The standard method for rat eradication on islands uses rodenticide anticoagulant poisons, typically brodifacoum or difficinon, distributed across the entire island's surface in bait stations or by aerial application. The precision required for aerial eradication operations is extraordinary. Helicopters equipped with specialized GPS-guided spreading equipment fly tightly spaced transects across the entire island, ensuring that every point on the surface receives a lethal dose of rodenticide. Bait must be available in sufficient quantity and for sufficient duration that every rat on the island encounters and consumes it. The operation must be timed carefully to avoid periods when certain non-target species, particularly birds, might be vulnerable to secondary poisoning from consuming poisoned rats or bait directly. The logistics of planning and executing a large-scale island rat eradication, particularly on larger, more remote, or more ecologically complex islands, can take years of preparation. Detailed surveys must map the distribution of rat populations and estimate their density. Non-target species must be identified and their vulnerability to the rodenticide assessed. In some cases, individuals of particularly vulnerable native species must be taken into captivity during the eradication operation and released after the risk period has passed. Whether windows must be carefully chosen, rain can wash away bait, wind can carry it into the ocean. Supply chains for the enormous quantities of rodenticide required must be established in advance. And then, after all that preparation, after the helicopters have flown their precise transects and the bait has been distributed and the weeks of waiting have passed, researchers conduct the most agonizing phase of the entire operation, the verification survey. They must confirm, beyond reasonable doubt, that every rat on the island is dead. This typically involves searching for signs of rat activity, footprints, droppings, burrows, gnaw marks across the entire island using methods designed to detect even a single surviving rat, tracking tunnels baited with attractants, wax blocks that record small mammal tracks, remote cameras. It is exhaustive, meticulous, and psychologically intense work because the consequences of missing a surviving rat population are severe. Despite these challenges, island rat eradication has become one of conservation biology's most reliable and successful tools. Hundreds of islands around the world have been successfully cleared of rats, cats, and other invasive mammals, and the results have often been dramatic and rapid. Native bird populations that were declining under invasive predator pressure have rebounded within years of invasive removal. Native plants that had been unable to set viable seed because rodents were consuming every seed produced have recovered as soon as that predation pressure was lifted. The ecological community, freed from the invasive pressure that was suppressing it, can demonstrate a resilience that is genuinely heartening. The recovery of bird populations after rat eradication can be almost explosive in speed, another demonstration of how fast evolutionary and ecological processes can move when the conditions change. Populations of seabirds that had been restricted to a few rat-free refuges on cliff faces have expanded onto island surfaces that were inaccessible under rat predation. Ground nesting birds that had been functionally unable to reproduce successfully because rats were taking every egg and chick have suddenly found themselves able to fledge young birds for the first time in decades. The species was there, the biology was there, the habitat was there, the rat was the sole obstacle, and removing the obstacle was sufficient to release the recovery. These successes have progressively scaled up in ambition. Early island eradications were conducted on very small islands, rocky outcrops of a few hectares where the logistical challenges were manageable and the risks of failure limited. As the technology improved and the experience of practitioners accumulated, operations expanded to larger and more complex islands. The eradication of rats and cats from the Antipodes Islands of New Zealand, covering approximately 21 square kilometers, was a landmark achievement in the early 2000s. The eradication operation on South Georgia Island in the South Atlantic, a sprawling mountainous island of nearly 4,000 square kilometers with a population of brown rats distributed across hugely challenging terrain, was, when it concluded in the mid-2010s, the largest island invasive eradication in history. The South Georgia eradication is worth dwelling on because of its scale, its complexity, and the extraordinary results it has already produced. South Georgia is a remote island in the South Atlantic, part of the same archipelago as Gough Island. Its rugged mountains rise to 3,000 meters, supporting glaciers that divide the island into discrete sections. Brown rats had colonized the accessible coastal areas of South Georgia, spread there from the 18th and 19th century sealing and whaling operations that had made South Georgia one of the busiest maritime industrial sites in the southern hemisphere. For more than 2 centuries, rats had been devastating the island's seabird populations, the same relentless predation of eggs and chicks that we documented on Gough Island, playing out on a vastly larger scale. The eradication operation on South Georgia required multiple phases over several field seasons, using enormous quantities of rodenticide delivered by helicopter. The glaciers that divided the island into sections were initially considered impassable barriers for rats, and therefore natural boundaries that would allow phased eradication. As the operation progressed, it became clear that rats were indeed crossing the glaciers in some areas, necessitating additional treatment of areas previously thought to be rat-free. The entire operation consumed extraordinary quantities of bait, helicopter time, and human effort, spread across seasons designed to avoid the breeding periods of vulnerable native species. The results have been transformative. Within years of the eradication's completion, survey teams began documenting bird species returning to breed in areas from which they had been absent for the entire era of recorded natural history on South Georgia. South Georgia pipits, the only songbird native to South Georgia, which had been restricted to small rat-free offshore stacks during the rat era, expanded rapidly onto the main island. Seabird populations began colonizing terrain that rats had made uninhabitable for ground nesters. The ecological recovery visible within just a few years of eradication completion speaks to the extraordinary resilience of island communities when the invasive pressure is removed. Gough Island itself has been identified as a priority target for rat eradication, specifically because of the devastating impact of mice on the island's extraordinary seabird colonies. The challenges on Gough Island are particularly acute because the target invasive is not a rat but a mouse, a smaller, more cryptic animal that is harder to detect and harder to deliver lethal bait doses to effectively. Mouse eradication has been successfully achieved on smaller islands, but Gough Island's 91 square kilometers, its complex terrain, and its exceptional ecological importance have made planning for its eradication a major undertaking. Conservation organizations have been working for years on the preparation and planning for a Gough Island mouse eradication, and the operation, if it succeeds, would represent a defining moment in the history of seabird conservation. The stakes on Gough Island cannot be overstated. The island holds globally important populations of the Tristan albatross, the Atlantic yellow-nosed albatross, the spectacled petrel, and many other seabirds. The Tristan albatross, in particular, is in a genuinely critical situation. Its population has been declining significantly, and mouse predation on chicks at Gough Island is considered one of the most significant factors in that decline. Albatrosses are among the slowest reproducing birds in the world. A breeding pair typically raises at most 1 chick every 2 years, and the chick takes many years to reach sexual maturity. When mouse predation is destroying a large fraction of the chicks that are produced each year, the population simply cannot sustain itself. The math of slow reproduction and high chick mortality does not work. Successfully eradicating mice from Gough Island would remove the most significant source of chick mortality that can be directly managed, allowing the albatross population to recover its natural reproductive success. Given the albatross's extraordinarily long lifespan, some individuals live for more than 70 years, even a partial recovery of chick survival could translate into significant population growth over the coming decades. The investment in eradication, though substantial, is completely justified by the potential population-level outcome. The broader lesson of island eradication operations, from the smallest rocky outcrops to the vast expanses of South Georgia, is that island ecosystems have a remarkable capacity for recovery when the pressure that has been suppressing them is removed. This is not universally true. Some island species have declined so far or have been so fundamentally altered by the changes in their environment that simply removing invasive predators is not sufficient for recovery. Some require additional active intervention, captive breeding programs, translocations, habitat restoration, supplementary feeding. But the baseline capacity for recovery is real, and it has been demonstrated enough times in enough places to be considered one of the most reliable principles in conservation biology. This capacity for recovery is itself a product of the same island rule that makes island species so vulnerable in the first place. Island species, shaped by evolutionary forces that selected for efficiency and specialization in stable environments, retain the biological potential to take full advantage of stable, predator-free conditions when those conditions are restored. Remove the rat, and the bird that evolved in a rat-free world can thrive again in the conditions its biology was designed for. The connection between the evolutionary history of island species and their conservation recovery is direct and profound. Nowhere is this connection more clearly illustrated, and nowhere has the science of island conservation been more intensively applied than in the story of the kakapo. This extraordinary bird, whose evolutionary history we outlined in the first part of this documentary, has become the focal point of one of the most ambitious, most technologically sophisticated, and most personally invested conservation programs in the world. The story of kakapo conservation is, in many respects, the fullest expression of what island conservation science can achieve when sufficient commitment, resources, and scientific creativity are brought to bear. The kakapo, Strigops habreptilis, was once abundant across both the North and South Islands of New Zealand and many of the offshore islands. Its fossils are found in deposits across the entire country, and the accounts of early European naturalists speak of encountering them in numbers. But the combined pressures of Maori and European hunting, forest clearance, and introduced predators, particularly stoats and cats, drove the kakapo to the brink of extinction with terrifying speed. By the 1970s, the only known wild population consisted of a small number of males on the Fiordland coast of the South Island. No females could be found anywhere. The species appeared to be functionally extinct. Then in 1977, a remnant population was discovered on Stewart Island, a larger island at the southern tip of New Zealand that included females. This discovery offered the possibility of a recovery, but it quickly became clear that the Stewart Island population was itself under severe predation pressure from feral cats. Despite ongoing efforts to manage the cats, kakapo continued to be killed faster than they could reproduce. The decision was made to remove the entire Stewart Island population to smaller, predator-free offshore islands where they could be managed intensively. This translocation, completed in the 1980s, was the beginning of what has become the Kakapo Recovery Program, a collaboration between government conservation agencies, research institutions, private donors, and a remarkable community of citizen scientists and volunteers that has transformed our understanding of this species and has pulled it back from the very edge of extinction. The numbers tell part of the story. When the recovery program began in earnest, the total known kakapo population was in the 60s. Today, thanks to the program's efforts, the population has grown to several hundred individuals, still critically small, but growing, and growing with a momentum that reflects the effectiveness of the science being applied. What makes the Kakapo Recovery Program particularly instructive from a scientific perspective is the way in which it has had to grapple with the specific biological challenges that the Kakapo's island evolved biology presents. Everything about the kakapo, every trait it developed over millions of years of island evolution in New Zealand, is simultaneously the thing that makes it extraordinary and the thing that makes it extraordinarily difficult to conserve. The kakapo's slow reproductive rate is the central challenge. Under good conditions, a female kakapo may only breed every 2 to 4 years, and she typically lays only a few eggs per clutch. This reproductive schedule was perfectly calibrated to a stable, predator-free environment where the key to reproductive success was quality rather than quantity, raising a small number of well-provisioned, long-lived offspring rather than producing large numbers of offspring annually as mainland birds do. In a predator-saturated environment, or in a population so small that random mortality events can significantly affect overall numbers, this slow reproduction is a devastating constraint. Making matters more complex, kakapo breeding is linked to the production of rimu, a native New Zealand tree fruit. In mast years, when rimu trees produce exceptionally large fruit crops, kakapo females respond to the nutritional abundance by entering breeding condition and initiating nesting. In lean years, when rimu masting does not occur, females may not breed at all. This reliance on an episodic and unpredictable food source for breeding readiness is another product of island evolution. In a stable, predator-free environment with a reliable long-term food supply, the adaptive strategy of concentrating reproductive effort in years of nutritional abundance makes perfect sense. In the context of a small population being managed for recovery, it means that breeding opportunities are infrequent and must be exploited as fully as possible when they occur. The kakapo recovery program has responded to these biological challenges with a combination of intensive management and cutting-edge science that has no real parallel in conservation biology. Every individual kakapo is known personally to the conservation team. Every bird has a name. Every bird carries a transmitter that allows its location to be tracked continuously. Every nest is monitored in real time with cameras and temperature sensors. When eggs are laid, they are weighed and candled regularly to assess embryo development. Chicks are weighed and measured at every stage of development. Females that are not producing enough milk, the crop secretion with which they feed their chicks, are supplemented with food directly at their nests. In breeding seasons, when females are actively nesting, conservationists provide rimu fruit supplementation, delivering food directly to nesting females to ensure they have sufficient nutrition to incubate their eggs and raise their chicks successfully. This intervention, which would be laughably impractical for any species that was not being managed at the level of individual birds with known identities and tracked locations, is practical for kakapo precisely because of the intensity of the monitoring infrastructure that has been built around the population. The use of genomic science in kakapo conservation has been particularly transformative. Because the entire population passed through an extremely small bottleneck, possibly fewer than 50 individuals at the lowest point, the genetic diversity of the modern kakapo population is severely reduced. Low genetic diversity in a small population creates real risks: inbreeding depression, in which the accumulation of deleterious recessive mutations in the homozygous state reduces individual fitness, reduced capacity for adaptive response to environmental change, and increased susceptibility to disease. Managing the genetics of the kakapo population is therefore as important as managing its numbers. Whole genome sequencing of every individual kakapo has been completed, giving conservation managers an unprecedented genetic map of the entire population. Using this information, breeding recommendations can be made that maximize genetic diversity, pairing individuals who are most genetically dissimilar to avoid inbreeding, preserving rare genetic variants that exist only in specific individuals, making decisions about which bloodlines are most important to protect. When a female enters breeding condition, researchers can select which male or males have the best genetic complementarity to her, maximizing the genetic health of her offspring. This is conservation genetics operating at its highest level, using the full power of modern genomic science to actively manage the evolutionary trajectory of a critically endangered species. The results of this genomic management are already visible. Kakapo offspring from carefully managed genetic pairings show measurably higher rates of egg fertility and chick survival than offspring from less managed breeding. The genetic health of the population, while still constrained by the severe historical bottleneck, is being carefully stewardshiped through every breeding season. Each new generation of kakapo carries a slightly optimized distribution of genetic variation, the result of thousands of hours of scientific analysis and careful management decision-making. The kakapo story also illustrates something important about the human dimension of island conservation. The level of individual care and attention that the kakapo recovery program provides would be unsustainable without the extraordinary personal commitment of the people involved. Rangers who spend months at a time on remote offshore islands, monitoring nests through the night in cold and rain. Veterinarians who perform complex medical interventions on individual birds, treating infections, rehydrating sick chicks, performing surgery when necessary. Geneticists who sequence genomes and run analyses to optimize breeding recommendations. Fundraisers, communicators, and citizen scientists who support the program with financial resources and public attention. This human commitment is itself a remarkable thing, a product of the deep fascination and emotional engagement that island species inspire in people who encounter them. The kakapo has a personality. Researchers who work with individual birds speak of them with a familiarity and affection that makes clear these are not abstract biological units. They are individuals with individual quirks and histories and relationships. The kakapo's unusual nature, its size, its flightlessness, its musty sweet smell, its extraordinary lifespan, creates an immediate and powerful connection between the bird and the people who encounter it. And that connection translates into commitment, into resource mobilization, into the sustained effort that conservation over long time scales requires. This is worth noting because it speaks to one of the most important practical lessons of island conservation. Charismatic island species, the kakapo, the Galapagos tortoise, the Komodo dragon, the albatross, serve as what conservation biologists call flagship species. Their extraordinary evolutionary histories make them objects of genuine public fascination. That fascination generates attention, funding, and political will that can support not only the conservation of those specific species, but the protection and restoration of the entire island ecosystems they inhabit. The kakapo's appeal as an individual, personable, deeply strange bird is an asset for New Zealand island conservation broadly, because protecting the kakapo means protecting the predator-free island habitats that support dozens of other native species that are less individually charismatic but equally important to the functioning of the ecosystem. The concept of flagship species and the broader strategy of using charismatic island species to drive broader ecosystem conservation is now a well-established part of island conservation practice. The Komodo dragon, as the emblem of Komodo National Park, draws conservation resources and international attention to a network of Indonesian marine and terrestrial habitats that support extraordinary biodiversity beyond the dragons themselves. The Galapagos tortoises anchor one of the most well-funded and closely monitored island conservation programs in the world, a program that protects not just the tortoises, but the entire remarkable Galapagos ecosystem. The kakapo anchors a network of predator-free island sanctuaries around New Zealand that collectively support the recovery of dozens of native species. But flagship species and eradication operations, as important as they are, represent only part of the toolkit of modern island conservation. Some of the most innovative and exciting work in island conservation biology is happening in the domain of ecological restoration, the attempt not just to protect what remains, but to actively recreate ecological relationships and community structures that have been lost. Ecological restoration on islands is grounded in a recognition that we articulated near the end of the first section of this documentary. Island species do not exist in isolation from each other. They exist in co-evolved webs of ecological interaction, relationships between predator and prey, between herbivore and plant, between seed disperser and the plants whose seeds they disperse, that have been shaped over millions of years and that are collectively responsible for the structure and function of the island ecosystem. Remove a key species from that web and the entire structure shifts. Restore the species and the web can begin to reassemble. The practical challenge of ecological restoration is identifying which species were key components of the historical ecosystem, understanding what ecological roles they played, and finding ways to either reintroduce them or replace them with ecologically equivalent species. This last option, using a closely related or ecologically similar species as a substitute for an extinct species, is called ecological substitution or rewilding with analogs, and it has produced some of the most fascinating experiments in island conservation. The most dramatic and best documented examples of ecological substitution come from work with giant tortoises. Giant tortoises, as we noted in the first part of this documentary, evolved independently on multiple island systems: the Galapagos in the Pacific, the Mascarenes Islands in the Indian Ocean, the Aldabra Atoll in the Indian Ocean, and various other islands. These tortoises filled an important ecological role as large herbivores and seed dispersers. They consumed large quantities of vegetation, maintained open grassland areas through their grazing, and dispersed the seeds of large fruited plants through their digestive tracts. When the giant tortoises of many island systems went extinct following human arrival, these ecological services disappeared with them, and the island plant communities began to change in ways that reflected the loss of their major herb. On the Mascarenes Islands, Mauritius, Réunion, and Rodriguez, the native giant tortoises went extinct following European colonization in the 17th and 18th centuries. The loss of the tortoises disrupted the seed dispersal networks of several large fruited endemic plant species, including some that appeared to be declining in recruitment, producing fewer young plants from seed, in the absence of an effective large-bodied seed disperser. Conservation researchers have tested the use of Aldabra giant tortoises, a species from the Indian Ocean that is closely related to the extinct mascaron species, as ecological substitutes on Mauritius. The Aldabra tortoise, introduced to a nature reserve on Mauritius, has been shown to consume the fruits of the large fruited endemic plants and disperse their seeds effectively through its gut. Germination rates of seeds passed through tortoise guts are actually higher than seeds that are not ingested. The physical and chemical processing in the tortoise digestive system scarifies the seed coat in ways that promote germination. The introduced Aldabra tortoises are, in effect, functioning as ecological substitutes for the extinct mascaron tortoises, restoring a coevolutionary interaction that had been disrupted for several centuries. This kind of ecological substitution is not without controversy. Conservation biologists debate the ethics and practicality of using non-native species, even closely related ones, in restoration contexts. There are legitimate concerns about unpredictable ecological interactions, about the risk of the substitute species itself becoming an invasive problem, about whether ecological substitution truly recreates the lost interaction or merely mimics its most obvious dimension. But in cases where the alternative is permanent loss of an ecological service that the island ecosystem depended on, and where closely related substitute species are available and well studied, the evidence suggests that ecological substitution can be a genuinely effective restoration tool. The Galapagos offers another remarkable example of ecological restoration that has both successes and cautionary lessons. The giant tortoises of the Galapagos were severely reduced by centuries of exploitation, primarily by mariners who loaded living tortoises onto sailing ships as a long-lasting food supply, a practice that removed hundreds of thousands of individuals from the archipelago over 3 centuries. Several tortoise species went completely extinct, and others were reduced to populations numbering in the dozens or even single individuals. The most famous individual is Lonesome George, the last known individual of the Pinta Island tortoise subspecies, Chelonoides abingdonii, who lived in captivity at the Charles Darwin Research Station on Santa Cruz Island from his discovery in 1972 until his death in 2012. Lonesome George became the world's most famous endangered animal, a symbol of extinction and conservation both, visited by hundreds of thousands of tourists and studied by researchers throughout his decades in captivity. Every attempt to breed him with females of closely related subspecies failed to produce viable offspring. When he died, his subspecies died with him. But the story of Galapagos tortoise conservation is not only the story of Lonesome George. It is also the story of extraordinary recovery efforts that have produced tangible results. The tortoise populations of Hispaniola Island, reduced to just 14 individuals by the 1960s, have been rebuilt through a captive breeding and reintroduction program to more than 2,000 individuals on the island today. The breeding program, operated at the Charles Darwin Research Station, hatches eggs from captive tortoises, raises hatchlings through their most vulnerable period when they are small enough to be predated by hawks and herons, and releases juveniles onto their home island when they are large enough to survive independently. Over 5 decades and thousands of tortoise releases, the Hispaniola population has recovered to the point where the population appears to be self-sustaining. Critically, the recovery of the Hispaniola tortoise population has also restored ecological functions that the tortoises provide, particularly the control of the vegetation structure on the island. Hispaniola had significant invasive plant problems in the absence of the tortoise population, with introduced species spreading into areas of native vegetation. As the tortoise population has recovered, the tortoises' grazing and browsing has begun to suppress some of these invasive plants and maintain the open habitat structure that the island's native flora and fauna depend on. The tortoise recovery is not just a single species success story. It is an ecosystem recovery, a demonstration that restoring a key ecological species can have effects that cascade through the entire community. The Galapagos more broadly has been the site of some of the most extensive and intensive island restoration work in the world. The removal of feral goats from Hispaniola, completed in 1978, was one of the first large-scale invasive herbivore removals in the Galapagos and demonstrated that island ecosystems could recover dramatically from even severe herbivore damage. Subsequent goat eradications on other Galapagos islands, including the massive operation on Isabella Island completed in 2006, have removed hundreds of thousands of feral goats from the archipelago, allowing the native vegetation communities to recover across enormous areas. The Isabella goat eradication is worth examining in some detail because of its scale and the innovative methods it employed. Isabella is the largest island in the Galapagos, covering more than 4,000 square kilometers of rugged volcanic terrain. Feral goats had been present on Isabella for decades, and their impact on the native vegetation was severe. They browsed native plants so heavily that large areas had been transformed from native forest and scrubland into bare, eroded terrain. The damage to the tortoise habitat on northern Isabella was particularly acute. The eradication operation, funded by a combination of international conservation organizations and the Galapagos National Park, used a combination of ground hunting and aerial hunting using helicopters. The aerial hunting component was particularly important on Isabella's northern volcano complex, where the terrain was too rugged for ground teams to operate effectively. Hunters working from helicopters were able to access areas that had never been systematically surveyed, let alone hunted, by ground teams. In the final stages of the operation, a technique called the Judas goat method was employed. Radio-collared goats were released in areas where eradication teams suspected residual animals might remain, on the logic that goats are social animals and a released Judas goat would seek out any surviving conspecifics, allowing aerial teams to locate and eliminate the last remnant groups. The operation took several years and removed more than 25,000 goats from Isabella and 2 adjacent smaller islands. It was, at the time of completion, the largest goat eradication in history. The recovery of native vegetation following the removal has been dramatic. Photographic time series from fixed monitoring points show the transformation from bare, eroded terrain to recovering native plant communities within years. Galapagos tortoises have expanded their range into areas from which they had been absent or at very low density, following the recovery of the vegetation they depend on. The ecological engine of the island system, freed from the suppression of invasive herbivory, has demonstrated the remarkable resilience that island communities retain when the disrupting pressure is removed. These successes in island restoration science, the rat eradications, the goat removals, the tortoise recovery programs, collectively represent one of conservation biology's most encouraging chapters. They demonstrate that the damage done to island ecosystems, while severe, is often not irreversible. Given the right interventions, given the commitment of resources and expertise, given the understanding of island ecological dynamics that conservation science has developed, island communities can recover. The evolutionary heritage encoded in island species can be preserved. The irreplaceable ecological relationships that have developed over millions of years of island isolation can be restored and maintained. But we would be giving an incomplete picture if we only discussed the successes. There are also failures and partial successes, in cases where the complexity of island ecological problems has exceeded the capacity of current conservation approaches. Understanding these cases is as important as celebrating the successes, because they tell us where the science needs to advance, where more resources and creativity need to be directed, and what the genuine limits of current conservation capacity are. One of the most challenging classes of island conservation problems involves what practitioners call invasive species that cannot be eradicated, species that are too widespread, too cryptic, too reproductively prolific, or too ecologically entangled with the native community to be removed completely. Cats, for instance, are notoriously difficult to eradicate from anything but the smallest islands, partly because they are secretive and wide-ranging, and partly because they are socially beloved by many of the human communities on inhabited islands. On inhabited islands, and many of the world's island species exist on islands with human populations, eradication of invasive predators is often politically and socially impossible, even when it is technically feasible. The conflict between conservation goals and the interests of human island communities is one of the most complex and sensitive dimensions of island conservation practice. Many of the world's most important island ecosystems are also home to communities of people who have lived on those islands for generations, who have their own deep relationships with the land and sea, who may not share the conservationists' view of which species belong and which do not. Effective island conservation in these contexts requires not just biological expertise, but genuine partnership with local communities, understanding their perspectives, respecting their knowledge and their rights, finding conservation approaches that align with rather than conflict with their interests and values. This is not always easy. Conservation programs that have been imposed on island communities without adequate consultation or partnership have a poor track record both ethically and practically. Community resistance can undermine even technically sound conservation operations. But conservation programs that have been developed in genuine partnership with island communities, that provide economic benefits through ecotourism or sustainable resource use, that respect and incorporate traditional ecological knowledge, have often been more successful and more durable than top-down expert-driven interventions. The story of community-based conservation in the Pacific Islands offers instructive examples of both the challenges and the possibilities. In many Pacific Island nations, traditional reef management systems, called taboo systems or rahui systems in various local languages, established seasonal closures on fishing in certain areas, allowing fish populations to recover. These traditional systems were disrupted by colonial-era changes to land tenure and governance, but there is now active work in many Pacific nations to revive and modernize these traditional management approaches, integrating them with contemporary marine biology to produce more effective and culturally legitimate conservation systems. On the terrestrial side, community-based conservation programs in island nations across the Pacific and Indian Oceans have engaged local communities in monitoring and protecting native wildlife, sometimes with remarkable results. Community rangers in Indonesia's Lesser Sunda Islands, which include Komodo's range, have become effective monitors of Komodo dragon populations and important intermediaries between the Conservation Management Agency and local fishing and farming communities. In Madagascar, community-based conservation organizations have been successful in some areas in reducing hunting pressure on endemic wildlife and protecting remaining forest fragments from clearance. These community-based approaches are not the dramatic technical interventions of a large-scale eradication operation. They do not make headlines in the way that a successful giant tortoise reintroduction does. But they may, in the long run, be the most important dimension of island conservation because they address the social and political dimensions of the problem that technical interventions alone cannot resolve. The ultimate survival of island ecosystems depends not just on the removal of invasive species and the recovery of native populations. It depends on creating conditions in which the people who live with those ecosystems value them, understand them, and have the capacity and the motivation to protect them over the long term. This social dimension of island conservation connects to a broader question that the study of the island rule raises about the human relationship with island environments. Why do islands matter to us? Beyond the scientific fascination, beyond the conservation imperative, why should the survival of a flightless parrot on a remote New Zealand island, or a giant tortoise on a volcanic Pacific archipelago, or a miniature hominin whose fossils lie in a cave on an Indonesian island, matter to people who live far from any of these places and may never see any of these creatures in their lifetimes? The answer, we believe, is both practical and philosophical. The practical answer is that island ecosystems, despite covering only a small fraction of the world's land surface, contribute disproportionately to global biodiversity. Islands contain roughly 20% of all bird species, a similar proportion of reptile species, and significant fractions of the world's endemic plant species, species found nowhere else on Earth. This biodiversity is not merely an aesthetic asset, though it is certainly that. It is a repository of biological information, genetic information, biochemical information, ecological information that has practical value that we have barely begun to explore. The compounds produced by island plants and animals, the ecological processes maintained by island communities, the evolutionary innovations that island isolation has generated, these have and will continue to yield insights with applications in medicine, agriculture, materials science, and environmental management. The philosophical answer is perhaps more important. Island species represent the products of evolutionary paths that are unique paths that, once ended by extinction, cannot be recovered. The kakapo has been traveling its particular evolutionary path for millions of years. Everything about it, its flightlessness, its extraordinary longevity, its slow reproduction, its nocturnal foraging, its complex social behavior, its musty sweet smell, is the accumulated product of millions of generations of selection and adaptation on the islands of New Zealand. No other lineage on Earth has traveled that same path. No other species will produce another kakapo. When the last kakapo dies, a unique evolutionary experiment, one that has been running for millions of years, ends. Forever, there is something that resists purely utilitarian accounting about this kind of loss. We are talking not just about the loss of economic value or ecosystem services or genetic information, but about the loss of a story, one of the most extraordinary stories that life on Earth has produced. The evolutionary story of island species, written over millions of years in the language of changing bodies and behaviors and ecologies, is part of the natural heritage of this planet. It belongs to all of us, not in a proprietary sense, but in the sense that the great works of human art and architecture and literature belong to all of us as part of the record of what is possible, what has been achieved, what the world contains. And when we allow that story to end through inaction or inattention or the failure to commit sufficient resources to its preservation, we are doing something that future generations will find genuinely difficult to forgive. Because the extinction of island species is not an inevitable consequence of human progress. It is a choice about what we value, what we are willing to invest in, what we believe we owe to the natural world from which we ourselves emerged. The island rule teaches us that evolution is creative, responsive, and remarkably flexible. Given the right conditions, isolation, time, ecological pressure, it produces solutions to biological problems that exceed anything we could have designed deliberately. It produces creatures so perfectly suited to their specific environments that they seem almost magical. It produces ecological communities so intricately connected, so precisely calibrated, that they function with an efficiency and resilience that modern engineering can only aspire to. Our task, as the species most responsible for the disruption of island ecosystems and most capable of their restoration, is to give that creativity the conditions it needs to continue, to protect the islands where evolution's most extraordinary experiments are still running, to restore the ecological relationships that allow those experiments to continue, and to ensure that future generations inherit a world in which the island rule is still operating, still producing wonders, still writing new chapters in the longest and most remarkable story that this planet has ever told. Let us now return to the scientific frontier, to the cutting edge of what researchers are currently discovering about island evolution and its implications for our understanding of life. Because the study of the island rule has not stopped advancing. If anything, the revolution in genomic and molecular biology has accelerated it, providing tools to ask questions about island evolution that were simply unanswerable even a decade ago. One of the most active areas of current research involves understanding the genomic basis of island adaptations, not just identifying which genes change when species evolve in island conditions, but understanding how those changes happen and how quickly they can occur. Researchers are now able to compare the genomes of island populations with their mainland relatives at a resolution that allows individual genetic variants to be identified and their effects on traits like body size, behavior, and physiology to be measured. What is emerging from this genomic work is a picture of island evolution that is both more complex and more elegant than previous models suggested. The genetic changes underlying island size evolution are not simple. They do not typically involve single mutations of large effect that suddenly make an animal dramatically larger or smaller. Instead, they involve many small changes across the genome, each affecting body size slightly, but collectively producing the dramatic size differences we observe between island and mainland populations. This pattern, multiple small effects adding up to a large phenotypic change, is called polygenic adaptation, and it appears to be the dominant mode of island size evolution. The advantage of polygenic adaptation, from an evolutionary perspective, is speed. When adaptation involves many genes, each contributing a small effect, natural selection can act simultaneously on many genetic variants, potentially producing observable change in body size within hundreds rather than thousands of generations. This is consistent with what we know about the time scales of island evolution in the fossil record, size changes that appear to occur over relatively short periods, at least by geological standards. The genomic architecture of island adaptation is itself an adaptation to the speed at which island conditions require evolutionary change. Another emerging area of research involves what scientists call epigenetics, changes in gene expression that do not involve changes in the DNA sequence itself, but involve chemical modifications to DNA or to the proteins around which DNA is wrapped that affect how genes are read and translated into biological products. Epigenetic changes can be heritable, passed from parents to offspring, and they can respond to environmental conditions much faster than genetic changes can accumulate through natural selection. Research on island populations has found evidence for epigenetic differences between island and mainland populations of the same species, differences in the patterns of gene expression that correlate with the ecological and physiological differences between the populations. This suggests that epigenetic mechanisms may be contributing to the speed of island adaptation, allowing populations to begin adjusting their gene expression patterns to island conditions even before the slower process of genetic change through natural selection has had time to operate. If this is correct, and the research is still preliminary, it has significant implications for our understanding of how the island rule works mechanically. It suggests that island adaptation may operate through multiple overlapping time scales, rapid epigenetic changes that allow immediate partial adjustment to island conditions, followed by slower genetic changes that consolidate and extend the adaptation over many generations. The island species we observe today may represent the end products of both processes working together over millions of years. The implications for conservation are also interesting. If epigenetic changes can occur faster than genetic changes, then the behavioral and physiological adjustments that island species make to changes in their environment, including the changes introduced by invasive species, habitat alteration, or conservation management, may be faster than purely genetic models would predict. Understanding the epigenetic component of island adaptation could help predict how island populations will respond to conservation interventions and could potentially be incorporated into management strategies. Another frontier in island evolutionary biology involves the study of microbiomes, the communities of microorganisms that live in and on the bodies of animals and plants, and that play crucial roles in nutrition, immunity, and various aspects of physiology. Island species, which have evolved in specific island environments for long periods, may have developed specific microbiome compositions that are as much a part of their island adaptation as their body size or behavior. The gut microbiome of a Galapagos tortoise, for instance, may be specifically calibrated to the plant communities of its home island, with microbial communities that efficiently digest the particular mix of plant material available there. If island species have highly specialized microbiomes, this could have important implications for conservation, particularly for captive breeding programs and reintroductions. Individuals raised in captivity or translocated between islands with different plant communities may lose their adapted microbiomes and experience reduced digestive efficiency or immune function as a result. Some conservation programs are now beginning to consider microbiome management, species recovery, testing whether supplementing captive animals with microbiome material from wild conspecifics improves their health and survival. The science is young, the applications preliminary, but the direction of the research is fascinating and potentially consequential. It represents another dimension in which the island rule, understood at ever deeper levels of biological organization, continues to offer new insights and new tools for both scientific understanding and practical conservation. The intersection of island evolutionary biology with climate science is yet another frontier that is becoming increasingly urgent. Islands are among the ecosystems most immediately and severely affected by climate change. Sea level rise threatens the very existence of low-lying coral islands. Ocean warming is bleaching the reef ecosystems that support island food webs. Changes in rainfall patterns are altering the vegetation communities on which island herbivores depend. And the changed timing of seasonal events, the masting cycles of trees, the breeding seasons of seabirds, the migration patterns of fish, are disrupting the ecological relationships that island communities depend on. For island species that have evolved in stable, highly specific conditions, climate change presents a particularly acute challenge. The kakapo's dependence on breeding readiness, for instance, is a trait that evolved when rimu masting was a reliable, if infrequent, signal of nutritional abundance. If climate change disrupts the timing or frequency of rimu masting, models project for the system in ways managers will need to actively compensate for. Similarly, the sea turtle populations that nest on tropical islands, themselves products of island-specific evolution, to natal beaches with extraordinary precision or thousands of kilometers of open ocean, face significant climate change. Warming sand temperatures affect the sex ratios of turtle hatchlings, since sea turtle sex determination is temperature dependent. Beaches that previously produced balanced sex ratios are now, in some locations, producing predominantly or even exclusively female hatchlings. If this trend continues, it could alter the reproductive capacity of turtles that have evolved over in the relatively stable thermal conditions of this. How islands climate change, management response, and challenges ecology tells us that islands have remarkable ability when but evolution time for generated tested by spread through the population. Happening fast adaptations with low genetic diversity. The question of whether evolutionary adaptation can keep pace with climate change is genuinely uncertain. This is where the conservation of genetic diversity within island species becomes critical not just for the present but for the future. A kakapo population with greater genetic diversity has more raw material for evolutionary adaptation to changed conditions. A Galapagos tortoise population that includes genetic variants associated with tolerance of higher temperatures or changed vegetation communities has a better chance of surviving a warmer future. Maintaining and, where possible, increasing the genetic diversity of island populations is not just about preventing inbreeding depression today. It is about preserving the evolutionary potential of those populations for the challenges of tomorrow. Some researchers have proposed more active interventions in the genetics of island populations facing climate change, deliberate introduction of genetic variants from other populations or closely related species that might confer greater climate resilience. This approach, called genetic rescue or assisted gene flow, is controversial and technically challenging, but in cases where alternative is extinction, it is increasingly being taken seriously. The principle is straightforward. If a population lacks the genetic variants need to adapt to a changed environment, and if those variants exist somewhere else in the species or in a closely related species, introducing them could provide the raw material for adaptation. The controversy around assisted gene flow centers on concerns about disrupting locally adapted genetic combinations. The specific genetic architecture that has made an island population well suited to its specific island environment may be disrupted by the introduction of genes adapted to different conditions. Getting the balance right between introducing genetic diversity and preserving local adaptation is a genuinely difficult scientific and management challenge. But it is a challenge that island conservation biology is engaging with, and the genomic tools now available to evaluate the risks and benefits was impossible even a decade ago. As we move to this documentary, stepping back tales of individual island systems and conservation programs to reflect on what they've taken as a whole, tells us about the nature of life and evolution on this planet. The island rule at its core is is responsive. Not consciously, not deliberate mechanism of differential survival and reproduction tracks its environment fidelity. Given isolate, it will reshape itself to fit whatever it will grow or new behaviors, form new ecologies, abandon new ways of existing in the world.